Hey guys, I'm just looking at the attendance report from yesterday. We had 63 people attend, so just wanted to share that with you guys.
Fantastic.
Yeah, that is an advantage of a virtual meeting, that there are people who are able to participate who wouldn't be able to participate in person. Yeah.
There's also the and of course the additional youth education component. The children that were in my car while I was driving home were forced to listen. So, yeah.
Yeah.
Margaret was I remember, you're listening to a professor from Manitoba right now.
Yeah, and it's amazing what they pick up. I remember when Sylvia was little, that she'd be coloring in the corner of the room, and I thought she wasn't paying attention, and then she's in the pool that night. Ichthyomyzon unicuspis, Geotria australis. And I'm just like, cool.
All right, I'm showing 5 minutes after. It is Friday, rumor has it. It's good to see everybody. Yesterday was fantastic. It's always exciting to hear from our HQP, and that's exactly what we did. I should probably introduce myself in case there's any folks that are from our so-called user community. I'm Stephen Cook. I'm a professor at Carleton, and I'm one of the co-leads of the GEN-FISH project. So yeah, I'm assuming, but that many of you were here for most of yesterday. It was really HQP focused. In the afternoon, we did some workshops, had some good discussions. Today, what we're going to do is start off with hearing about all of the themes that we have. So we have 5 different themes. And then later today, we've got some sort of open but somewhat structured discussions that we will have focused around these themes and other issues. So today should be this morning's a bit more of a brain dump, and this afternoon is more of an interactive session. Nonetheless, there's certainly opportunity to ask questions, and there was pretty robust use of the chat yesterday. We're able to save that and certainly use that, so encourage that as well. I know that it's a lot of sitting, and so there are a couple breaks built in, but trust that everybody's able to manage their mind and body themselves and get up and walk around. At some point, I'll probably go for a walk and throw it on my cell phone and listen and view that way. So there are ways to do this without requiring a chiropractor next week. So with that, I think we're going to jump right in. I think 1 and 2 are going to be done together. And just have to toggle back. And I am seeing that's yesterday. Today, so yeah, 1 and 2. Specific names aren't listed here, but you know who you are. I think we're looking at probably Margaret as Margaret, and I don't know if Nick Mandrak's here or not. I can't see him, but
yeah, presenting on activity 2.
Okay, great. All right, take it away.
Okay, let's see how the share screen works. Is that good?
Looks great.
Yep.
Okay, let me just, oops. I've lost everybody's faces. There we go. Now I can see faces as well. Yeah, so I'm Margaret Docker at the University of Manitoba, and the third of the 3 co-leads on GEN-FISH, and the Activity 1 lead. And so it's really exciting to be able to give what I think will probably be a flying overview of our plans and progress to date on Activity 1. So Activity 1 is the development of the FISH survey, the eDNA toolkit. So just to sort of give you an overview of what our very ambitious objectives were for this, it seemed like a good idea 4 or 5 years ago, but certainly is keeping us busy. So sort of the cornerstone of Activity 1 is Objective 1.1. developing those early stage validation eDNA assays for all freshwater fishes of Canada. So that's 198 native species plus 28 invasive species that are either already here or sort of at the doorstep that are of concern to managers. And then this consists of, we'll be developing single species assays, targeted assays for using 2 markers. So there's some redundancy there for all species. And then I'll also talk a little bit about our efforts to do sort of more community-wide or sort of panels of assays with open array chips and metabarcoding. Objective 1.2 was the environmental microsatellite DNA, then rather than using a single species specific marker, using amplifying microsatellites from water samples to see if they can be used for quantification. And then the third objective was stomach content DNA for predator-prey analyses. And so I think this is probably the only slide that shows the PIs. All the rest will focus on the HQP, who are by and large doing most of the work, so that this activity involves 6 PIs from 6 universities, coast to coast, and a really exciting collaborative project. So just a very, very quickly, so that when I'm talking about the assay design and validation, there's the 3 basic steps: in silico, which is the computer-based part, where we design and predict the assay specificity based on gene sequences of the target versus the non-target species. In vitro, which literally means in glass, so that's in a tube, when we use tissue-derived DNA to then test how well those in silico predictions work in reality. And then the third step, the in situ or in place validation, where we test this on environmental samples where we know the species is present or absent. That the large scale in situ validation and testing will be happening in activity 2, but I'll talk about a few of the small scale in situ validation, just to make sure that what's working in the lab works as expected in the field. And one of the things we'll be finding as we go through is a lot of these, the initial design and validation, of course, requires comprehensive reference sequences to design the primers and reference tissue collections to test them. And this is part of what we've been accomplishing in this activity as well. So just very quickly then, so in terms of the in silico validation, the computer-based testing, when we started, we naively thought that there were a number of published assays that we could more or less adopt wholesale for Canadian freshwater fishes. So Stephen Rogers, one of Bob Hanner's students at the University of Guelph, did some in silico testing of 101 published CO1 assays and found, let's see what it. Here, let me see if I've got the numbers here. He found only 5% of the assays were truly species specific. Another 20% or so were regionally specific. They would at least work in one region or another, but not universally. And so a lot of his work then, he came up with this nice sort of decision tool where you can sort of work through this this decision tool to determine whether, blue, the assay is truly species specific. Green, it's regionally specific. Or in the case of the red or the purple, maybe it will give you false positives, detecting something else but not your species, or false negatives, where maybe it will miss the species of interest. This was disappointing, but it was certainly necessary for us to know this before we moved forward. So then what we've had to mostly do then is, given the sort of underwhelming performance of the published assays, at least for our purposes, design for the most part all of our own assays from scratch. During the eDNA roundtable discussion yesterday, Brooklyn Litke gave a nice overview of the process. I loved her figure here, how we like to think, especially if you're a novice to the eDNA field, that oh, I'm going to design an assay and then next week I'm going to use it sort of thing. And that there's an awful lot that goes on between the deciding to use eDNA and detecting eDNA with your assay, and quite a few steps involved. And just to sort of quickly sum this up, and so that what you're wanting to do is have enough sequence for from your target species so that you can design an assay that will work for any individual or any population, and enough sequence from non-targets to make sure that your assays don't amplify DNA from other species in the same genus, the same family, or the same geographic area. And this is where we depend a lot on existing sequence alignments. And so a lot of our initial efforts were put into gathering publicly available sequences, or in some cases, generating our own to fill in gaps. And then there's a lot that goes on in terms of designing assays that work well for a given type of assay, for a particular thermocycler, for example. And there's a lot of expertise that we're developing within GEN-FISH to be able to do this efficiently and effectively. And so this, as I think you're probably starting to see, was and is a huge team effort trying to design TACMAN assays for those 2 mitochondrial genes for all freshwater fishes of Canada. So again, these are some, I hope I've got most, if not all, of the HQP who have been involved, sort of, again, coast to coast, from UNBC to UNB, designing and validating these assays. So I believe that the count right now, that of the 226 Canadian freshwater fishes, we have 212 TACMAN assays for CO1 and 31 for Site B. The reason why we have so many more for CO1 right now than for Site B is there's just much more sequence available for CO1. So we were able to start more easily with that. And so this is one of the things that we're really trying to develop and contribute as a sort of an end product of GEN-FISH, something that will be shared with the entire community when we're done. Right now, everything is just on Teams for internal GEN-FISH use. Paige has been really working to help keep this up to date and complete, where all of the different GEN-FISH team members can keep track of all of their assays, the sequences, and so on. And then one of the things that's really important, and I'll talk about this a little bit more when we talk about validation and reporting requirements, recording all of the really essential information. So as I said, that very few published assays are truly species specific, so you have to at least be able to qualify what you mean when you say it's species specific. Is it species specific in Manitoba? Is it species specific in New Brunswick? And so recording what species might be potentially problematic based on in silico analysis, what species have actually been tested against the assays in vitro. And so all of this information is well documented and available, so that moving forward, individuals can take a look and say, oh, okay, I see it cross-reacts with Atlantic salmon. Atlantic salmon doesn't occur in my region. I'm good to go, and those sorts of things, so that we aren't having end users aren't having to sort of go blindly forward when they're using these tools. And so even though it's going to be some time till we have it all 226 species compiled and ready to share publicly, sort of collections of assays are starting to be published now, usually based on sort of regions. And so Rena at the University of New Brunswick, one of Scott Pavy's master's students, finished her thesis in the summer. She developed 20 Site B assays for New Brunswick freshwater fishes. Her thesis is, I believe, now publicly available, and a manuscript based on this will be submitted to PLOS ONE very soon. And again, one of the key things for the trustworthiness of these assays is that all of this information be made publicly available, so that she's provided lots and lots of supplemental tables that provide all of the information that you need. And so looking at this, for example, when you look at Atlantic salmon versus brown trout, how many mismatches are in each of the primers and the probe to really get a sense of the reliability of these. And then other sort of regional assay collections that a number of my students at the University of Manitoba have been developing and testing eDNA assays for a lot of Manitoba fishes. And so those will be shared again, sort of as a package as we're working toward that big release of all 226 species. Second part of the toolkit is the eDNA metabarcoding. You heard about this from Ian Smith yesterday, and this is sometimes referred to as community eDNA, so that instead of assaying for a single target species with highly species-specific primers, you try to use very general primers that will amplify everything, and then you use high-throughput sequencing to be able to then identify the species of interest. And so the first step was to, again, like we did with the targeted assays, was to test the usefulness of existing primer sets for Canadian freshwater fishes. So Erica Myler at the University of Guelph. This was part of her master's thesis that she finished this past year, testing 2 existing CO1 primer sets against conventional sampling and historical records. And in particular, the third chapter of our thesis was looking specifically for taxonomic blind spots. And taxonomic blind spots are consistent false negatives for specific taxa. So where you're missing specific taxa using the eDNA barcoding. And the potential reasons for these are either inefficient binding of so-called universal primers. Maybe those universal primers aren't truly universal and they're missing some species. Another reason for a taxonomic blind spot are gaps in the reference sequence library. So you're sequencing the species, but you can't match it up to anything because there's a gap in the reference sequence library, or insufficient resolution between species. That with closely related species, if you have a very small fragment of eDNA, maybe there just aren't sufficient differences to resolve them. Somewhat surprisingly and certainly disappointingly, she found approximately 50% of the species in her collections were affected by taxonomic blind spots. So eDNA was capturing, some of the same species that were found in electrofishing and in the historical records, but certainly missing a lot as well. And so this is where GEN-FISH comes in the same way that with the targeted assays, we had to design a lot of our own. A large team at the University of Windsor in the Heath Lab is trying to redesign and improve those metabarcoding fishes, particularly focusing on their application to Canadian freshwater fishes. So a couple of the things that they're doing then to improve the ability for these primers to work on Canadian freshwater fishes to minimize those taxonomic blind spots is to, first of all, use longer amplicons. That will help improve resolution, presumably. If you have a bigger chunk, you're more likely to be able to distinguish between closely related species. Improving primer sequences to hopefully get more universal primers and to fill in those gaps in reference sequence libraries. So just as an example, these are some redesigned 12S primers going across a diverse range of taxa. Looking pretty good for all teleost and even non-teleost bony fishes, so like lake sturgeon, for example. Where it's not maybe 100% great, that sea lamprey, which is very distantly related to the others, was amplifying, which was surprising, but maybe not quite as strong. So you're probably catching all of the taxa, but maybe with some differences in frequency. So that's definitely seems to be an improvement over the existing ones. And part of this will also be testing on eDNA mock communities. Linda Al-Rafay at the University of Windsor doing her honors project with Dr. Daniel Heath, and Matthew Yates is trying to develop and test these new and improved CO1 metabarcoding primers. And what's really exciting here is if we look here at the sea lamprey, we see really good amplification across all taxonomically diverse Canadian freshwater fishes. So that'll definitely minimize those taxonomic blind spots. And Linda will also be doing this mock community testing, spiking DNA from tissue-derived samples into Detroit River water to see whether, you get out what you put in, and having these DNA pools from 20 species, again, to test the ability to do that community eDNA barcoding. And then the sort of the third part of this is something that wasn't originally planned in the Genome Canada proposal, but seems like a really good opportunity, is to use an open array eDNA chip to sort of find that to hit that sort of middle ground in between the single targeted assays where you're assaying for one species at a time, or the entire community with metabarcoding, that you can have assays for 28 species arrayed on a single chip that you can then run in duplicate. And this is building off of Matt Sharon's stomach content eDNA work that he did for his master's. And so now we're sort of testing some of these chips to see how well they work. And I just realized that I'm getting up to my time limit, so I'll definitely pick up the pace here. And so then the third part of this then is the in vitro testing, the lab testing to see how well those designed primers perform in reality. And then an important part then is the standardized reporting requirements. So to do this in vitro testing, we're relying on an extensive reference tissue collection that in the first couple of years of GEN-FISH, we put a lot of time into building up this collection with a lot of vouchered specimens from throughout their geographic range in Canada. And again, this is something that this reference tissue collection is on teams trying to make sure that we have location information for all of the samples so that we know exactly where things came from, and in many cases, the source of the sample to verify identification. And as with our other protocols, I mean, definitely we'll be sharing them all at the end, but we're trying to share as much as possible as we go through. And so we already have a tissue or a DNA request form that's on our GEN-FISH website. Right now, it's largely for internal purposes, but the idea is to make as much of our information broadly available. And in terms of protocols, trying to develop various protocols that can be adopted by end users. One of the things that we did early on was just looking at the literature to see whether there was some standardization in eDNA protocols for fish surveys, and we found definitely that wasn't the case, as you can just see here quickly. All sorts of different collection protocols, filter types, filter pore sizes, filter preservatives, extraction methods. I mean, it really made us very aware how, say, novice eDNA users who might want to adopt eDNA, how quickly overwhelmed they would be if they went to the literature. And so part of what we're trying to do then is to make that information much more accessible. And so, again, this is something that we've already shared with a number of our partners, particularly those who are doing field collections for us, but we're also trying to make as much of it possible available on the website. Even if it's just available in draft form, there are various videos available to show some of the sampling protocols. And we're continuing to refine that, especially with feedback from partners. And then the in vitro testing, where we use that reference tissue collection to see how well the assays perform in reality, and developing protocols for specificity. Does it only amplify the species of interest? Sensitivity, what's the limit of detection, and whether compounds in the environmental samples might interfere with the PCR. And so then this is the HQP have been really busy over the last several months, leading on trying to standardize, develop a paper to report this standardized protocol. And one of the things that was really nice here was, I mean, that coming up with this assay validation protocol with recommendations for both the in silico assay design and the in vitro and in situ testing. But really trying to make sure that it was sufficiently streamlined so that it would remove some of the barriers that typically limit the widespread adoption of eDNA, but still making it rigorous. And so that there needs to be that balance where the results are sufficiently trustworthy, but you aren't sort of letting the perfect become the enemy of the good, where that there is such stringency required that maybe you never get to the end point. And then they use 3 case studies to sort of illustrate from beginning to end how you would validate an assay, whether you were adapting and validating a published assay, whether you were designing and validating a genus-specific assay, or designing and validating a species-specific assay. So the hope is that this is really useful then for people who want to get into the eDNA sort of business, but maybe are a little bit intimidated by some of the 80-page documents that go through some of this. And again, I won't talk very much at all about the in situ testing, that the large scale in situ testing is part of activity 2, but in pretty much all of the studies that developed and did the in vitro validation of eDNA assays, they also did some initial in situ testing where they knew whether the species was present or not to make sure that the assays worked in reality. And this, I'm including this too because it was generally the most fun part of the project, because this is where the HQP had an opportunity to work with some great partners. And then again, I just want to quickly mention that in the course of developing all of these tools so far, we've also had to sort of generate a number of other resources that I think will be useful to the community as well. And so, for example, that just this year in 2023, Mandrake and Al chapter on the zoogeography of the freshwater fishes of Canada came out. And so we're using this to put together updated species lists that, first of all, update the taxonomy for all freshwater fishes of Canada. So there's a number of family and genus changes that our lists now sort of quickly convey, whether something is native in most of Canada, but invasive in parts of Canada, invasive to Canada, invasive but a low priority, invasive but a high priority. So these are lists that may be of use to others in the community outside of GEN-FISH. The distribution of different species and different major drainage basins, their glacial refugia. These are again lists that might be broadly useful. And also because a lot of the work that we're doing is meant to have conservation and management implications, we also have the conservation status and links to reports for a lot of these species. And these are things that we hope to also share at the end. And then I'll just very quickly mention the other 2 objectives that we've had some really exciting developments in the testing of environmental microsatellite DNA for quantification. And so that this project, this objective was led by Dr. Brian Neff at Western and Dan Heath as well. And so I'm just going to mention some of the work that the MSc student Simone McCloskey did at Western. She finished that up this summer, trying to test whether amplifying microsatellite loci from water samples could be used for quantification of Atlantic salmon. So tested very nicely, sort of in the lab, in stream enclosures, and then in situ in 3 Lake Ontario tributaries. Found that microsatellite loci could be amplified from environmental samples, but unfortunately they could, the genotype of fishes couldn't be reliably gotten from that information. And they think that it was probably because with sort of the degraded environmental DNA, that there may have been a sufficient amount of sequencing errors that genotype couldn't be determined. But what was potentially valuable still is that we always struggle with the fact that our other assays are based only on mitochondrial markers, and so then this would give nuclear eDNA markers if needed, particularly for well-studied species where microsats exist or are known. And then the last thing I'll mention is the stomach content DNA work. And I think, if I'm remembering correctly, that the application of this will be part of activity 2, but in terms of the development, this was part of Matt Sharon's master's work, and he tested both the ability of metabarcoding and an open array chip to identify stomach contents of 5 predator species. If you've ever done stomach content analysis, you probably realize that visual diet analysis often won't allow you to distinguish different prey species, and so this is where DNA is particularly helpful to be able to identify what species might be in that digested mass. And so using metabarcoding, and so then just quickly to orient you to what we're looking at here. So each vertical line is a single stomach sample, and then the different colors within that are different prey species. And so you can see that the metabarcoding allows multiple species to be identified within each of the stomachs. And then for even faster processing of stomach contents, using an open array chip to be able to assay for 28 key prey species simultaneously. And Matt and Daniel have recently submitted a manuscript based on this to molecular ecology resources. Oh, and then the one thing I will add, what was quite surprising here in the 9 assays where qPCR TACMAN assay was compared to the open array chip, the sensitivity was similar between the 2 approaches. The assumption has always been that an open array chip might be a lot more efficient, but would have lower sensitivity because of the small volumes of material. But to find similar sensitivity to the TACMAN assays was quite exciting. And that's it. And I apologize if I've run over.
Thanks, Margaret. So Matthew, do you want to go? And then if there's time, we'll have questions after your presentation. If not, there's the afternoon for discussions. You're muted, Matthew.
How about now?
I can hear you.
Perfect. Can we see the screen?
We see your team screen overlaying your. There you go.
There we go.
Oh, it's
well,
back.
I messed it up. I don't know. Interesting. Yeah, there we go. All right, I clicked the wrong button. I'm assuming you guys see all this, and there's no little Teams window in the bottom corner.
It's perfect.
All right, wonderful. Okay. Yeah, so my name is Matthew Yates. The activity lead for activity 2 is Dr. Nick Mandrak. Unfortunately, he's not able to be here today, so I'm going to be presenting in his stead to just describe the progress that we've made on activity 2, which has 2 real kind of main objectives. The first is sort of a broad scale eDNA survey where we assess fish communities in 500 plus water bodies across Canada, and with objective 2.2 focused on sort of more lower level case studies that were predominantly co-productions actually with GEN-FISH folks and other sort of government or non-academic agencies. So let's talk about objective 2.1 first. So the first main task of this was just to collect the samples itself. So we went across Canada and collaborated with academics, government agencies, and non-academic agencies as well, or groups as well, to sample more than 500 water bodies for eDNA samples. And this has largely been completed at this point. So we've made pretty great progress on that. We're now fully into task 2.1.2, which is applying the eDNA TACMAN assays and metabarcoding assays described by Dr. Docker previously to start quantifying or obtaining data on fish communities from these samples. And an objective 2.1.3 will be to compare the historical and eDNA fish data for these water bodies to assess sort of the reliability and variability observed in eDNA sampling relative to traditional sampling. So for the first objective 2.1.1, the collection of eDNA samples across Canada, we've actually made pretty good progress, solid progress on this. In the last 3 years, between 2020 and 2022, we got a pretty wide range of samples across the country. Notably, we're lacking samples from northern regions. They're pretty difficult to collect. 2023, we were planning on really focusing sampling efforts on obtaining samples from northern regions and sort of supplementing with a focused effort on that particular region of the country. But unfortunately, most of our sampling efforts for that fell through for a variety of reasons, including forest fires and a variety of other calamities. But we're going to try again next year to sort of help supplement with that samples from the northern regions of the country with, again, targeted sampling. So we're optimistic that we'll be able to get that. We also have commitment from the provincial ministry in Quebec to provide a number of samples from across Quebec itself. So we're in the midst of 2.1.2 currently, and I'll get into that later in the presentation. But so now, for now, I'm going to switch to objective 2.2, where I'm going to discuss case studies. And there's 3 different sort of sets of case studies that we're looking at here. The first being applying eDNA and environmental microsatellite DNA to specific case studies. 2.2.2 is applying stomach content DNA methods, and 2.2.3 will be kind of the sort of wrapping it up comparison across case studies to identify methodological challenges that emerged across all of them. So these case studies, like I said, are actually really good examples of what Dr. Cook was talking about yesterday, in that most of them were actually co-production efforts with GEN-FISH academics and government or non-government agencies, where we looked at conventional, in a lot of cases, looked at conventional sampling to compare against eDNA sampling, or looked at eDNA sampling or stomach content DNA sampling to derive ecological information on species and communities of interest. So the first one I'm going to talk about, well, actually, one thing I want to mention is one thing that's been very interesting for me anyways, is watching, I know all of these case studies from the beginning of my work with GEN-FISH, but now we're actually starting to have a lot of data generated for these case studies. And I'm going to focus a lot on discussing some of these preliminary results for these different case studies that are actually starting to produce data. So the first one I'm going to talk about is a pelagic species survey in the Williston Reservoir. And this is led by the Shrimpton Lab, where they used eDNA to monitor pelagic fish communities, where they looked at things like community composition, species distribution, and also the correlations between eDNA and fish abundance. And I think they had a lot of focus on developing novel qPCR methods, but also employed metabarcoding assays to look at this, and looked at eDNA concentrations and detections across multiple depth strata. And preliminary results from this are, unsurprisingly to me, interesting, because they seem to demonstrate a positive relationship between the concentration of eDNA observed in a lot of these systems and relative abundance of these fish species, and in case of qPCR assays and eDNA concentrations, the absolute abundance of these fish species. You can see that fish species that tend to or the concentration of eDNA in these environments tend to reflect the relative biomass of these of the different species for which qPCR assays have been developed. And so there's a couple other case studies that I'll just briefly mention coming out of the Shrimpton lab. They've got one where they're looking at enumerating, they have a counting fence on rivers in the BC, and they're going to be counting fish that move upstream or downstream, sorry, and looking at the correlation between eDNA and fish abundance for different groups of salmonids. And so this is an ongoing project, but the Shrimpton lab also added a new case study recently where they're going to be looking at using eDNA to differentiate closely related sympatric species. British Columbia streams tend to be dominated by two groups, the salmonids and Echodidae, and the Shrimpton lab will be developing novel qPCR assays for species from these groups and collecting eDNA where these species occur in both sympatry and allopatry. And so again, this is an ongoing project. So I'm going to switch now and move a little bit further east to talk about some projects undertaken by the Docker lab. One of the case studies in GEN-FISH is to map the distribution of endangered carmine shiner using eDNA. Because they want to monitor range changes as a result of contractions due to habitat loss, pollution, and potentially climate change. So they developed a series of eDNA assays, or 2 eDNA assays, and successfully deployed them to examine the distribution of carmine shiner. And they found consistent detections within the historic range distribution of these fish. As you can see here, red dots represent a positive detection. And interestingly enough, they also ended up finding that there wasn't a drastic difference between detection rates and different filtration methods used to collect it, with possibly vacuum filtration performing a little bit better than the other two, but overall, somewhat consistent results. The Docker lab is also looking to use eDNA to map the distribution of 6 species of freshwater fishes of conservation concern in Manitoba. So they're conducting widespread sampling of water bodies across the province, focused on 6 different key species. And they shared some preliminary results with me, which look pretty interesting. We've got the preliminary results for bigmouth buffalo that actually suggest eDNA detection suggests that bigmouth buffalo are occurring outside of what is thought to be their historical range, and we see similar patterns for golden red horse as well. You can see the historical range outlined by the yellow on this figure here, and some novel eDNA detections that are putatively outside of this historical range. So that's, again, another example of the efficiency and effectiveness of using eDNA to map species distributions. And finally, I'm going to talk about, out of the Docker lab, another case study where they looked at using eDNA to detect deepwater sculpin in very deep freshwater lakes. Deepwater sculpin are kind of a glacial relict species at a lot of different lakes. They're a species of special concern because they have very narrow niche requirements. They depend on very deep, well-oxygenated water, so there's a limited number of water bodies that they can really inhabit. And they're also very difficult to detect with traditional methods, as you can appreciate, because they tend to inhabit extremely deep water and they're fairly small fish. So the Docker lab used eDNA to detect or to look for deep water sculpin in 2 very deep freshwater lakes in Quebec, and they've successfully detected eDNA at all sites sampled in 31 Mile Lake, but unfortunately didn't detect any sites in Lake Pemichigan. But my understanding, I think, is that they're going to go back for some further supplementary sampling to see whether or not they can kind of investigate the possibility of them inhabiting Lake Pemichigan further. But again, this kind of highlights the effectiveness of eDNA in a lot of situations where we have traditionally difficult to detect species. And now we're going to switch to some projects focused in central Canada. This is a project coming out of the Mandrake lab where a master's student, Markel Morfett, used eDNA to classify fish communities in agricultural drains in southwestern Ontario. And they paired eDNA sampling with traditional sampling. And so we were able to generate some results with that. And again, eDNA sampling was overall largely consistent with environment or with traditional sampling, but this does highlight the importance of controlling for contamination. We process some of these samples, and you can see here at Gleer where we have lots of Chinook salmon. Chinook salmon are showing up in pretty much everything. So these results led us to kind of tighten up our contamination controls. And I'll speak to that later on as well. But one thing that actually I just even realized, these results are interesting because this is something I even only realized last night after seeing Ian's presentation, but these kind of results and tying across these different projects can be very useful for highlighting potential problems and identifying potential problems. You can see here that black bullhead are identified in a lot of these pond or agricultural drain environments, and brown bullhead are notably absent from most of them. We would expect a fair number of brown bullhead, I think, in a lot of these environments. So coupled with Ian's finding that DFO found brown bullhead, but black bullhead were showing up in a lot of his eDNA samples, I suspect that either there's something going on in terms of hybridization that Dr. Heath mentioned yesterday, or there's a potential misidentification in terms of the reference sequences that were posted on GenBank that we use to identify these individuals. So that'll be something interesting to investigate moving forward. And so there's a number of other case studies occurring here in Ontario, like looking at eDNA, the effectiveness of eDNA metabarcoding to estimate the abundance of entrained larval fishes. And this is an ongoing project that sampling for this project is ongoing, but my understanding is that there's a fair number of results that indicate that metabarcoding is actually relatively effective or reflects the relative abundance of these larvae in these bulk samples. Similarly, the Mandrake Lab is also undertaking a project to compare conventional and eDNA methods to quantify nearshore fish community composition and health in the Saugeen Peninsula. And they've undertaken a novel project. Again, Markel Morfett, a student out of Dr. Mandrake's lab, will be comparing lake whitefish and round goby eDNA concentrations with traditional sampling. And this is important because round goby, which are an invasive species, could be a potential predator of lake whitefish eggs, which are socioeconomically important fish species. And so this is sampling that's currently undergoing, and we'll hopefully have important results, or the implications of this study will have, or the results of the study will have important implications for the management of fish species important for collaborating Indigenous communities with this study. So I'm going to switch now to some stomach content projects. These are projects, applied projects coming out of Objective 2.2.2. And so this is Matt Sharon's study. You heard Dr. Docker talk about the different assays that were tested and validated for this species, and there were quite a number of them. You can see pictured here, a large number of assays went through a variety of tests to examine the effectiveness of their sensitivity, specificity, and interference, most of which passed. Only the ones highlighted in red were ultimately excluded. Matt Sharon applied these assays to examine stomach contents of 5 Great Lakes predator species, and he got some really interesting results out of this, I think, that indicated that these fish or sort of revealed the ecology of a lot of these different fish species in terms of their prey preferences. They definitely demonstrated some ontogenetic diet shifts in the feeding ecology of these predator species, with walleye, for example, showing a switch from smaller prey like gizzard shad to larger prey items like rainbow smelt as they got larger, as you would expect. And we also found some interesting results associated with freshwater drum. Freshwater drum were thought to shift to a benthivorous, they're piscivorous at early life history stages, but as they get larger, they shift to benthivory. But we ended up finding that even as they are benthivory focused on sort of kind of with a more focused consumption on invertebrates. But we ended up finding that they didn't really switch completely to invertebrates, and they remained piscivorous because they just ended up feeding pretty much exclusively on round goby as fish species prey. And I think that's interesting because that's kind of a novel finding regarding the ecology of freshwater drum. And Dr. Mandrak will also be engaging in a novel study examining or identifying predator or prey species, stomach contents of key fish species in Lake Huron to examine the impact of predators on potential fisheries species and look at their trophic interactions. But this project is kind of sort of currently ongoing, and he's looking for a postdoc to start analyzing these samples. And again, another case study coming out of the Mandrak lab looking at using ichthyoplankton samplings to detect invasive and endangered fish species. And this was a largely successful project that has actually been, the results of which have actually been published, where they've ended up finding that they were able to detect a lot of different species using this method, including 3 at-risk species and 3 invasive species. And another project or sort of sub-project involved in this case study involved comparing larval fish sampling and sequencing to optimize methods. And they ended up finding that, importantly, the taking a larger number of samples was actually more important than generating very deep sequencing, because there was a lot of interreplicate heterogeneity in terms of the species detected within a sample. So more samples, better than deep sequencing, which is really important for these types of metabarcoding studies. And finally, I want to talk about, or not finally, one more case study after this, but Dr. Doctor mentioned the use of environmental microsatellite eDNA to look at signatures of abundance in Atlantic salmon. This is coming out of the NEF lab in collaboration with Dr. Heath. And again, they ended up finding some preliminary results indicating that there was good correspondence between the number of individuals detected and the eDNA that they recovered in natural streams. And finally, now I'm going to talk about another case study coming out of the McIsaac lab, led by Ian Smith, master's student Ian Smith. You heard from him yesterday, actually. This project focused on Hamilton stormwater ponds, many of which were drained to provide absolute census, and the rest of which were on which DFO implemented traditional census estimation methods to obtain data on the fish communities present and compare it to the data obtained from sampling eDNA. And Ian actually has some really interesting results. Again, when we focus on a specific species, problematic invasive species in the area, which are goldfish, preliminary qPCR data indicates a positive relationship between goldfish abundance and the concentration of eDNA recovered from these ponds. And metabarcoding data generated from metabarcoding, data generation from metabarcoding is currently ongoing, as you heard from Ian yesterday, but we're hopeful, we're optimistic. He seems to be getting some pretty interesting and good results, but that data has to be sort of processed and cleaned a little bit further before we start making key inferences from it. And finally, I'm going to talk a little bit about some of the SOP or standard operating procedure optimization that we've been undertaking at GLIR as a part of Activity 2. We've been developing a lot of lab protocols with a focus on cost effectiveness and efficiency, because sometimes there can be a trade-off between these two variables. So for Activity 2, we've trialed a large number of methods to extract and extract eDNA, including different homogenization methods like using investigator baskets, Kaya shredders, or bead-based homogenization, and different extraction protocols, including using commercially available kits like QIAGEN DNase kits versus sort of in-house sucrose lysis or digestion buffer. In-house created sucrose lysis buffers or digestion buffers. And we ended up settling on a protocol that included investigator baskets for sort of a homogenization step, and our sort of custom digestion buffer that we make in-house. And this provided a good balance of sort of DNA recovery and cost efficiency and scalability. You can see that using QIAGEN shredders here, we're provided a little bit better of a yield, not that much better, but they are substantially more expensive and way more labor intensive than the method that we ended up choosing. So this is what we ended up settling on for the large scale survey, for processing the samples in the large scale survey. We ended up excluding robotic extraction. This was something that I mentioned last year that we were going to trial, but our tests showed that it was unfortunately a bit too, not necessarily inefficient, but inconsistent in terms of the recovery of eDNA, and it posed a potential contamination risk. But we did use the protocol that was essentially developed for the robot and just and use that to develop a sort of high throughput protocol that could be performed by a technician, except instead of having a robot arm perform the extraction, we had a technician arm perform the extraction. And it actually performed quite well. We're getting very good results from this. And we've now processed through most of the filters, the vast majority of filters collected for the broad scale survey, and are applying the metabarcoding assays and soon, hopefully, open array chips for the various TACMAN assays that have been developed for Canadian freshwater species through Activity 1. And finally, I just want to highlight that our perspective piece on the challenges of applying eDNA for fisheries management and conservation in Canada was published in CJFAS. And so that's out now. That was a collaborative effort across a large number of labs and PIs in GEN-FISH. And we also were able to successfully obtain a CFI GELF grant led by Dr. Heath to fund the establishment of a dedicated eDNA laboratory at GLIR. So this is now up and running, and we were using this in the summer to do this sample processing for Activity 2. And I also just want to mention as well that through the EGF, GEN-FISH labs and partners can submit samples to it for a sort of fee-for-service arrangement to be processed in this dedicated eDNA facility, where we very sort of tightly control what can and cannot enter this laboratory, and have very sort of strict, clean protocols for sampling or for processing only eDNA samples in this dedicated room. So that's it for me. Yeah, I'd like to conclude with that. And I guess I don't know if we have any time for any questions, either for Dr. Docker or myself, but well, Dr. Heath, I guess.
We started about 5 minutes late, so I think there is time for a few questions.
I've got one if no one else does. I'm just curious how exactly you spike DNA in a river like the Detroit River.
I can handle that one if you want, Dr. Docker, because I'm helping with
I can
that
guess,
process.
but I will let you, so I don't have to guess.
Yeah, so basically what we've done is our undergraduate student has extracted DNA from tissue samples from 20 different species that are sort of broadly representative of taxonomic groups of Canadian freshwater fish species. And then she also went into the Detroit River, collected eDNA samples and extracted them in the eDNA lab. And then we're basically spiking in this tissue-derived DNA, diluted of course, into the eDNA samples. And the purpose of that is to make sure that the DNA is, the markers that we're validating using these sort of mock communities and our mock eDNA samples are still effective in the presence of things like inhibitors and potential interfering DNA. Because we did have, for example, we developed two 12S markers exploiting the ion torrent, the ability of the ion torrent to generate larger amplicons. And both of those markers performed very well on just raw tissue extracted DNA. But then when we actually went to apply them to environmental DNA samples, one of them did not perform very well, actually. And we ended up discarding that during the developmental process and selecting only the one that seemed to show consistent results both across tissue and across actual real eDNA samples. So yeah, we found that validating using real eDNA samples is actually a pretty important step for these metabarcoding primers anyways.
Gotcha, thank you.
Joe pointed out that Anne-Marie has a question.
Oh yes, thank you. So thank you both of you for your presentation. That was excellent, and it's awesome to see that there are some great tools that are being developed or that are already developed. So as I say yesterday during the eDNA session, in Pass Canada, in national office, actually, we are making a document, an internal guidance document for making eDNA as a measure into the monitoring program. And because we cannot handle everything, because we are working in every ecosystem types in Pass Canada, we have decided to focus on freshwater fish species. So making a document, especially for implementing eDNA measures in freshwater fish species monitoring. So I would like to hear from you how we can make use of the tools you are developing into this protocol or guidance document. So, and the main point maybe is that in Pass Canada, we do not have any genetic lab. So it means that everyone in Pass has to deal with a collaborator. So the document is just a guidance document for every step that they have to conduct for monitoring freshwater species with eDNA. So I'm sure that we can make use of your tools and just integrate into the document, or I don't know how to do that, but I would be happy to hear what you think about that.
Yeah, I think we talked about this a little bit yesterday, how, we want to be able to share what we're developing as widely and as soon as possible. And the balance, of course, is making sure that we aren't sharing things prematurely if they aren't fully validated. That can do more harm than good. What might be helpful is, like I was indicating, I mean, we've got sort of packets of assays that are being published as part of theses or as part of manuscripts that as they're developed. At the very least, I mean, we could share with you theses as they're published. They're publicly available, but you wouldn't necessarily be looking for them if you didn't know that they were there. So that knowing, for example, that Stephen Rogers at the University of Guelph, he's done a lot of that in silico analysis. So that might help you know which of the published assays in your database you might need to stay away from and which ones you can test. Rena's thesis is published, that we can sort of start that way. It's hard with trying to compile an entire list because we just aren't there yet for some of them. On a species by species basis, sometimes we'll have partners contact us and say, do you have an assay yet for white sucker? And we might have it almost there, but because it's just for one species, we can then take a look ourselves and say, okay, what species are in your area? Okay, yeah, no, this one I think is good. We just don't have that done yet for all of them. But I would say at the very least, starting with published theses, and then at some of those papers as they're being rolled out, that at least gets you going sort of incrementally before we're ready to sort of release the whole thing. Because it would be a shame if we've learned over the past few years that certain published assays can't really be used in Canada, but we don't share that information, and then so others then repeat the same mistake or whatever. So being able to sort of share what we get as we get it, at least, would be an interim step, I would say.
Yeah, definitely. Thank you.
Thank you. I think in the interest of time, we'll move on to the second toolkit, starting with Ken talking about activity 3. Ken?
Looks good.
Oh,
Good, Ken.
excellent. It's a little bit of a delay. Thank you for the introduction, Daniel. Okay, so as Daniel just said, I'll be talking about activity 3, which is the development of the Fish Health Toolkit. Okay, so the main deliverable for activity 3 is to produce what we are calling a universal TACMAN qPCR array. And so we've kind of narrowed that down to calling it the stress response transcriptional profiling chip, or the STP-chip. And our original goal at the start of this project was to produce a 112 qPCR assay gene chip, STP chip, that could be used for as many different species as possible. That has proved to be quite challenging. But we are still producing these STP chips that work for as many different species as we can. So it's kind of focusing in on groups of species at this point. And so the approach was to first, through discussions with other PIs associated with activity 3, was to compile a target gene list. From there, we started out trying to compile the transcriptomic resources for the target genes for our target species. And so this was either done through downloading gene sequences from publicly available databases, or through using reference transcriptomes that we've produced in our own labs for other projects, or in some cases for more understudied species, to go out, get samples, do some RNA sequencing to produce our own reference transcriptome for having the gene sequences for our target genes. And with that information, we could then align the sequences together for the different species and try to design our qPCR assays within the conserved regions of those genes. And so from there, we could start producing these STP chips and then testing them out on as many different species as possible. As I mentioned, the original plan of doing 112 genes for every freshwater fish in Canada proved to be difficult. And so we've started producing a lot more of these, what we're calling specific STP chips. And these are 56 gene chips that are typically targeting processes that are associated with gill, muscle, and liver, but they will work for other tissues as well, or a brain-specific chip, because there are a lot of processes that tend to be specific to the brain. And we've now started covering a lot of the major groups of species that are relevant to freshwater fisheries in Canada. And so when we started out doing this, we focused on salmonids first. So the chars, Pacific salmon, Atlantic salmon, and what am I missing? Oh, the whitefishes as well. And so we started out with trying to produce a 112 gene chip for the for all of the salmonids, and this involved compiling sequences from in collaboration with 7 different laboratories within GEN-FISH. The assays were designed by folks from 4 different laboratories. And so we were trying to target sort of these genes that are associated with these types of physiological processes. And so this was largely led by Dr. Shahin Islam, and he's been absolutely instrumental in a lot of the progress that's been made for activity 3. And so from this, we were actually able to produce what kind of a database of different assays that that worked for different species of salmonids. And from that, we can start producing these more specific 56 gene STP chips. And if we start branching out to the other groups of species, we have about 14 different STP chips that have been produced now associated with activity 3. And since the last AGM, we now have this system set up at the University of Manitoba as well as at the University of Windsor, so we can start processing samples quite a bit faster that way. And then I think one of our big problems that we're going to have to face moving forward is to start standardizing the analysis of the chip data. But that'll come as we're now starting to produce a lot of the data associated with activity 3. And again, now that we have these chips produced and we're starting to run different samples on them and start to compare across species, we're starting to generate a lot of the data that's going to be associated with some of the progress in activity 3. So we have a lot of papers in the work, and I'm very optimistic that we'll see a lot of published results coming out of activity 3 over the next year. And so one of the studies that we had done just to start trying to test out and validate this multi-species approach, this was done by, again, Dr. Shahin Islam. And what he did was he tested out the performance of that 112 gene STP chip for salmonids, and he tested it out on 8 species from 4 different genera. And he was able to test that out on multiple tissues for all of those different species. So we were able to then compare the performance of those assays across the different species. And remember, these are qPCRs that have been designed in the conserved regions of our target genes. And then now we're applying that same qPCR assay to multiple species. And this is just a little bit of a teaser of the data that have been produced. There's not a whole lot that you can take from this, but for all of those 112 genes, there were a lot of genes that showed up that they're able to amplify across all 8 species. And then depending on the tissue, the number changed a little bit, and there's still going to be some of the assays on that original gene list were more specific to other tissues like the brain. But this is a PCA plot of the 67 genes that amplified for all 8 species in the muscle tissue. And essentially, there's not a whole lot you can take from this, but you do seem to see that a lot of the species cluster together quite nicely. And where you start to see a little bit of a difference along PC2 is where you have the bloater and the lake whitefish tend to be separating out, which is not terribly surprising because they would be a little bit more diverged than the rest of, or less closely related than the rest of the salmonid species. But you do see that the species tend to cluster within each other quite well. So we've got rainbow trout here, we've got Arctic char, Atlantic salmon. So it works well. We get consistent results for within a species. We tend to have a little bit of a different pattern between the species, which would be expected. And so one of our challenges moving forward is going to be how are we going to incorporate the differences in the performance between the different species into our analyses. I guess it's not a challenge, it's just something we'll have to address. And then since our last AGM, another study where we were able to sort of test out these STP chips across multiple species. This was done where we developed a 56 gene chip for Accipenser sturgeon. So if you don't know what a sturgeon is, or I guess this group, everyone would, but this is a lake sturgeon here, and they're just a wonderful group of species to work with. And we just developed a 56 gene chip for quantifying thermal and general stress responses, and then also looking at food deprivation. And this was done largely by Hossein Hajiji and Kyle Madden. And what we were able to do with these chips is then to test out the performance of the chips on the liver and gills for 4 species in relevant to freshwater in Canada. And we were able to then show that the assays that were designed tend to work quite well. We did get amplification for all the species for most of the genes. We're going to have to redesign a few of them for version 2 of the chip, but that's going to be part of the process as we produce these chips, figuring out which ones work well, and then try to optimize and redesign and just make a final product that works all the time for the different species. But this is going to be a really useful tool for looking at this group of highly endangered species. And I just from personal experience, this particular chip has got a lot of interest from several research groups across Canada. And so one of the things that kind of came from this is we were able to start producing this large database of assays. And so we know that we have these assays that work for different species. So then from there, we've started being able to produce these kind of what we're calling sub chips, or these more specific chips. And so the salmonids are are the ones that we have the most STP chips for, and we have specific ones that are designed to look at various types of environmentally relevant processes. And we're already at a point now where we're starting to redesign and optimize and get into second iterations of some of these chips. And so we've produced a lot of different of the specific STP chips for salmonids. We have our ones for sturgeons, but we've also started to produce ones for perciforms and as well as for cyprinids. And we've just started now. We're in the process of designing ones specific to sea lamprey and hopefully other lampreys as well. But this activity 3 tends to be quite technical. What's really exciting about this is how it can be applied to various types of biological questions, and that's associated with activity 4. So at this point, I'm going to turn it over to Daniel for an update on activity 4.
Thanks, Ken. See how sharing my screen goes.
You're good.
That worked?
Yes.
Yeah, good. Okay, so I'm going to be talking about activity 4, which is sort of the validation of the chips that are being developed in activity 3. The process of validation comes under 3 objectives. The first is sort of the laboratory validation, and that's headed by Nick Bernier. The second is validation of the chips under field conditions at multiple spatial scales. So this is under field conditions, but known or at least suspected stressors. That's led by Céline Audet and Christine Semenuk. And finally, there's the objective 4.3. This is proof of concept. This is sort of real world applications in partnership with some of our end users. And that's led by Trevor Pitcher and Steve Cook. So I want to remind you, I've shown this slide a number of times, that the point of activity 4 is to go beyond the in silico in vitro validation or development of the STP chips and target in sort of more live testing or validation using known stressors, but also moving on to situations where the stressor may be suspected but not known. So put the objectives on this continuum. Objective 4.1, this is lab-based or tank-based, where we're applying single or multiple stressors, but we know what the stressors are and we can control for other unanticipated stressor effects. Then we move to the field-based challenges, where we may know what the applied challenges we're providing, but we don't actually know if there's some synergistic effects that are associated with the field or more natural mesocosms. And then the final is the partner initiated. This is where partners come to one of the members of GEN-FISH and say, this is a problem we're having. Can we use one of your chips to see if that's going to help us with this particular problem? Bear in mind that this is often associated with systems where there are multiple stressors, some known, perhaps some suspected, or perhaps some stressors that they have no clue about being at play. So objective 4.1, this is controlled stressor validation using both single and multiple stressors. And I'm just going to give you a really quick update of where we're at on these 3 different objectives. So what you see here is a table. Along the y-axis, we have the species. Along the x-axis, we have the stressor. Note that I've actually combined a number of different water quality issues under water quality. Our original proposal had about 45 species that were targeted for the STP chip and about 10 stressors, which leads to 450 single stressor assays alone that had to be done. And then we get into multiple, and the number of combinations and permutations of two stressors for multiple stressors just became mind-boggling. And so we pitched to the Research Oversight Committee, and they were accepting that we're going to cherry pick, we're going to choose species stressor combinations that make sense and not try to saturate this diagram. But even so, we're actually doing relatively well in terms of the diversity of species and the diversity of single stressors that we're using. Remember that these black bars represent studies that are in some level of production. So it may be just initiated, it may be tissue in the freezer, or it may be data from the STP chips. If we look at the multiple stressor, it's a more limited range of projects. This, of course, gets a lot more complicated in terms of actually designing and doing the studies. But even so, we're starting to look at this potential for nonlinear effects of multiple stressors. We go to 4.2. Again, this is where we're applying stressors. So it's a controlled, and I use those air quotes limitedly as far as I can, but it applies here. So we are applying controlled stressors, but we're in field or semi-natural systems. And those are where we're trying to see whether the outcomes from the lab-based are fulfilled when we go into a more natural system. And again, this is somewhat more sparse than the single stressor projects, but we can see that there is a diversity of species that we've done field mesocosm or gradient studies, and a real diversity of stressors. What I would like to point out about Objective 4.2, a lot of these projects are at the planning or tissue in the freezer stage. There are not a lot that have progressed to the point of actually generating qPCR data. Objective 4.3 was identified by our ROC, the Research Oversight Committee, and the original committee that we talked to when we were trying to get money for GEN-FISH as being really important. And this is more for trust building, as came out in the discussions yesterday, where we're not taking systems that we're very comfortable with and saying, look, it worked, but rather we're working with partners or end users, and they're providing us with the challenge, and we're using our STP chips to see whether or not it solves the problems that are meaningful and in a solution that is useful for our end users and partners. Because of the excitement surrounding the potential for the STP chip, sort of this fish health toolkit, we actually have 21 projects that are in some level of development, some of them at the point where they're actually generating qPCR data. But look at the diversity of species and the diversity of stressors or project descriptions, ranging from things such as stocking evaluation, so the effect of tracking stocked individuals, to hypoxia and thermal stress in natural systems. So we can see that there's a lot of interest, and I could easily see this list doubling in the next year as our chips come online. And from my experience anyways, that partners are contacting me with questions about, could we use a chip for this? So this is really exciting. Okay, we talk about chips a lot. There are a few of us who handle the chips, and we know exactly what's going on. But for many of you, probably it's sort of a black box. So I'm going to take 2 minutes here and describe what the heck is a chip. You've probably seen this image on lots of the GEN-FISH talks. This is an actual open array chip that you buy from Thermo Fisher. You can notice with this blow up that there are a whole bunch of little dots on the chip. Each of those are through holes, and the sides of those through holes have all of the reagents necessary for one individual qPCR assay. So every one of those dots is like a little tiny test tube to do a qPCR assay to determine the gene transcription level. And the pattern of this is not random. There are actually 48 of those subarrays, or those squares of dots. Those squares of dots are 7 by 8, so 56 dots. So when you hear Ken talking about a 56 gene array, that means that those 56 dots have 56 different genes printed on them. So how do you apply this? Well, you take cDNA from fish number 1, and the machine will robotically put that cDNA in all 56 of those holes in that first subarray, all the way through to fish 48. So this is why we keep telling you, if you want to run a chip, you need 48 fish, and you're going to get data for 48 fish, 56 genes. So each one of those fish is going to be assayed at 56 genes. Now, if we're interested in a fairly narrow set of genes, let's say osmoregulatory stress, we can actually design it to be 28 genes, but then each gene is replicated. That makes the 56. When Ken talked about the 112 everything chip, that actually is 2 chips. So you print the first 56 genes on chip 1 and the second 56 genes on chip 2, and then fish 1 has to go on chip 1 and chip 2 to get data for all 112 genes. So if you're doing a project where you're using a 56 gene or 112 gene array, you're going to have to run multiple chips for each fish. So that's just to give you an idea of what the constraints or design parameters are for the chips, because at this point, we're going to start getting partners coming to us saying, we want a tox chip. And then we dive into the 112 gene, and then Ken can design one for us. Okay, this is what leads to all of these specialty chips that Ken described. And these are what leads to the activity for validation. Every time we design a new chip, we need to come up with some form of validation on real live fish. The first step could be just as simple as saying, are the assays working? So we just grab cDNA from whatever the target species is and say, do all of the genes actually produce something? But that's very preliminary. The next step is going into the lab, into the field, or working with a partner to actually try it out. If you are partnering with an end user in the validation 4.3, it's really critical that we make it clear to them that they're part of the validation project. This is not going to solve, well, might, but we can't guarantee it's going to solve their problem because we're still in the validation stage. But that means it's the co-production, and they're going to tell us if they're happy or not. And that's critical for, say, activity 5, barriers to adoption. Okay, now I'm going to give some selected examples, and this is just to highlight what's going on in activity 4. A lot of these examples you saw yesterday because the HQP described their own projects. And so what I'm doing is sort of concatenating all of those together to show you how it fits into objectives 1, 2, and 3. So objective 4.1, this is the laboratory validation. We have lots of examples on that, so I'm only going to do 3 that highlight 3 of the different types of objective 4.1 studies. So one of the ones that came out of interest from our end users that was not in our original proposal, so this is sort of extra, is non-lethal sampling. We originally planned that STP chips would involve killing the fish. You have to kill them and get a liver sample or a brain sample or a muscle sample, spleen, whatever it might be. But the idea was, okay, you're going to have to sacrifice the fish. Many of us got feedback saying we want to do it without killing the fish, particularly if we're dealing with a fish that is a species at risk, which makes perfect sense. So a number of people have worked on non-lethal sampling trials. I know Nick Bernier's done some, Steve Cook's working on some of these. The one I'm going to describe is work done by Shahin Islam et al. And this is looking at osmoregulatory stress and non-lethal sampling for transcriptional profiling of Chinook salmon. So what Shahin did is partnered with Yellow Island Aquaculture, who are being forced to move from saltwater rearing, which you see behind me, to all freshwater rearing of Chinook salmon. They're interested in markers for osmoregulatory stress that might affect the growth or performance of their fish. What Shahin did is he set up this replicated design using freshwater controls, because these were 20-gram fish that had not yet been in the ocean. So the fresh water is basically staying where they were. Brackish as they moved to about 15 part per 1,000, and seawater is full strength seawater. Shahin then sampled the fish at 0, 12, 24, and 48, 72 hours. He's generating the data even as we speak, except I think he's on this call. But if he wasn't, he would be generating the data as we speak. And the non-lethal samples, but requiring handling, was a blood sample, a skin swab, and a gill biopsy. So in these cases, you have to catch the fish, but you don't kill them. You take non-lethal. Shahin also sampled for environmental RNA in these tanks, because if it's going to work, it's going to work in this situation. And of course, once he did all this on the live fish, he bumped them off and did a traditional lethal sampling of liver and gill. And here we see a similar PC plot to what Ken showed. This is just preliminary data, but you can see the red is the brackish water, the blue is the fresh water, and the green is the salt water. On the right, we see lethal sampling of liver for messenger RNA. We can see a really nice delineation amongst the treatments along PC2, less delineation along PC1, but there is, pardon me, I've got that reversed. PC1 showed delineation, PC2 showed a little less. I'm getting confused, but you know what I'm saying. Interestingly enough, if we go to the non-lethal gill biopsy, there's still a significant differentiation in terms of the transcriptional profile between the three treatments, but it's much more subtle. We don't know whether this is related to the fact that it's a tiny sample and we're dealing with limited amount of messenger RNA, or whether it's just a different tissue that's responding differently to the challenge. And it might be that the gill is remodeling in a very rapid way, so it's showing less challenge effects. We have to look at earlier challenge times to see if we have this temporal effect. But this highlights the fact that we've got this potential to do non-lethal sampling and still get transcriptional profiling. And Shahin's working on the eRNA, and I don't know about any of you, but I'm holding my breath to see if the eRNA actually provides a transcriptional profile that reflects the challenge.
Yeah, we are getting a promising data, Daniel.
Fantastic, Shahin. Thank you. Just wait to the next AGM that's going to be in Turks and Caicos. We'll invite you down, Shahin. Okay, controlled single stressor trial. This is the one that we have lots of examples, and the example I'm going to describe to you is work done out of Mark Shrimpton's lab in Ken Jeffrey's lab, working with bull trout. So this work was done by Carol Best et al. And Carol, I was given this picture of you drinking beer in the north by Ken. So anyways, as we know, the bull trout is a native species of Western Canada, and they're declining. They're of serious concern. It's been theorized that they're declining because of increasing water temperatures associated with climate change. And it was recognized by Ken and others that thermal threshold data can really inform recovery efforts to identify populations that are at risk because of peaking temperatures or elevated temperatures in general. The design that they used was these bull trout were reared at 10 degrees C, and they were moved into divergent temperature treatments ranging from 6 to 21 degrees. They were left there for 3 weeks, and then they were assessed. The endpoints to assess thermal tolerance included classical physiological tests such as critical thermal maxima, etc. But also notice that they did the transcript abundance, that is, doing transcriptional profiling using gill, liver, and muscle to see whether it could be more sensitive than classical critical thermal maxima type measurements of thermal tolerance. And these are their data showing acclimation temperature on the y-axis, CT max. I'm getting my X and Y's mixed up. Acclimation temperature on the X and CT max on the Y. And this is pretty classic thermal tolerance plot where you see the breakdown in terms of the ability of the bull trout to tolerate thermal or acclimate at 21 degrees C. When they looked at their transcriptional profile data, they saw a common pattern of changes in gene transcription across gill, liver, and muscle with some of the genes. And you can see both up and down regulation and some unchanged. Interestingly enough, when they used the principal component plot to highlight this pattern, you can see this very obvious divergence among the challenges as you move down PC1 to the 21 degree treatment, indicated that you're getting metabolic challenge and oxidative stress. The interesting thing, as reported by Carol, is that at 21 degrees, the bull trout are classically showing thermal tolerance breakdown. And this is perhaps not surprising. They're cold water species. 21 degrees is pretty high. But the take-home message, at least for GEN-FISH from this project, is the transcriptional profile data started identifying changes associated with thermal tolerance breakdown at temperatures at 18 degrees C or even lower. So this provides perhaps a way of detecting thermal stress or thermal tolerance breakdown prior to getting to the point where the fish are actually dying. Okay, and the last one for objective 4.1 is controlled multi-stressor trials. This is the microbiome manipulation study by Javed Sadeghi. He gave that talk yesterday. And again, I'm going to go through this quickly because I'm highlighting this as one where you're going in there and you know there are multiple factors affecting the gene transcription profile of the fish. So as Javed explained yesterday, we know that there's this complex interaction between the gut microbiome and the host gene expression, well, the host in general. And while it is a back and forth thing, as far as GEN-FISH is concerned, we're really interested in what happens when you change the microbiome in terms of the host response to that. We know that if you modify the host microbiome, you can affect the health both positively and negatively. And so the design that Javed used were these replicated tanks where the fish were either fed a normal diet, a diet supplemented with probiotics, or the same feed supplemented with antibiotics. The analysis consisted of two steps. There's more to it that I'm not going to go into a lot of detail about, but one is he selected a single family for RNA sequencing to identify candidate genes for the transcriptional profiling chip. But then he did all the fish using the transcriptional profiling chip, and this was a 56 gene array. So he had to run two chips for every fish to get his duplicate, his replicate. And as you may remember from his talk yesterday, there were subtle but significant differences in the gut microbiome of the fish that either got the antibiotic treatment, that's in red, or the blue, which is the probiotic treatment, or the green, which is the control, no treatment, normal feed. So we know that the treatment he did affected the microbiome, but what about the fish? Of course, Javed has a lot of information. The one that I'm going to talk about is actually the correlation between the abundance of specific bacterial taxa with the expression of gut tissue genes. And you can see that 4 of these correlations were statistically significant. They were all negative correlations, which meant so when lactobacilli went up, the expression of those 2 genes went down. And I'd also like to take 1 minute to have a bit of basic personal rant. about the tyranny of Bonferroni. This was come up with by a bunch of ecologists, not transcriptomists, where you can have thousands of simultaneous tests. Because notice that, in fact, for certain groups of bacteria, we're actually seeing relatively strong positive correlations with gene expression, but it's not statistically significant because of the FDR corrections that are necessary. But in my opinion, way too conservative for the type of data that we're dealing with. That's the end of my rant. Now I'm going to go into objective 4.2. These are the field validations. So these are mesocosms or gradients. And there are a number of these projects that have started. Very few of them, as maybe none of them, have got to the point of actually generating data. I may be incorrect on that. But the one I'm going to talk about is a student in my lab, Patricia Voye. And she's got tissue and is in the process of generating data very soon. So Patricia was interested in environmental stress gradients, and she's focusing on both temperature and sort of nutrient pollution gradients. She presented this in a 3-minute talk yesterday, so I'm repeating. But the interesting thing about her project is she included sort of a natural gradient of temperature in the Bay of Quinte, where over a fairly small geographic range, you have a large change in temperature. And also in Lac Saint-Pierre tributaries, we have populations that are experiencing high agricultural impact or low. So more specifically in the Lac Saint-Pierre tributaries, there are some that are basically almost pristine and forested, and it ranges all the way to highly agricultural impact. And she sampled 3 different species there, identifying 3 different trophic levels. In the Bay of Quinte, she focused only on yellow perch, but she was able to sample yellow perch from deep in the Bay of Quinte, where the temperatures are quite warm, to more closer to the bay, to the mouth of the bay, where you're having the effect of Lake Ontario and the temperatures are lower. What she's been working hard on is developing the chips that are relevant for this thermal / nutrient pollution gradient. And so she's been working and has completed the Persiforms chips with the genes that you see listed to the right. This has been tested and looks like it's going to work. I believe, Patricia, you're at the point where you're actually generating data, and she's at the process of testing the candidate genes, so for the same chip, but targeting cypriniforms. And at this point, it's looking good, and I keep encouraging her to order the chips because there's a 6-week delay. No data yet, but next time we'll get to see. Okay, so I'm moving to objective 4.3, and I'm going to give 2 examples, then I'm done. The first one is, so these are partner-initiated projects, and sometimes it's hard to decide whether your project is a 4.2 or a 4.3, and the criteria that I ask people to use is, would you have done this on your own? If this was something you say, I want to do, and you go out and grab the partners, then it's not the same thing, because you're going to choose a project that has high likelihood of positive outcome. But if OMINRF comes to you and say, look, we're transporting Atlantic salmon for reintroduction, and we're worried that transportation is really decreasing their chances of success, and they say, Trevor, can you do something about it? That's when it's a partner-initiated project. So this work is headed by Dane Roberts et al. And again, he gave his 3-minute talk yesterday, so this is nothing new, but I'm putting it in context of objective 4.3. So what Dane is doing is looking specifically at the transport stress in the hatchery-reared Atlantic salmon. He's also looking at some other things, if you remember his 3-minute talk, but I'm targeting the transport stress. He provided this slide that I really like, and the problem is transport is unavoidable when you're reintroducing fish. And the transport results in not one stressor. Transportation by itself is not a stressor. It's all that other stuff. So during transport, you have problems with water quality, food deprivation, injury, all of these things that are listed on this slide. So it's truly way beyond any of our multiple stressor kind of designs. It's kind of a black box. And so identifying what level of stress is present and what you could do to try to reduce that stress is critical for OMNRF, who's interested in maximizing the success of the reintroduced salmon. So the plan is to sample the fish at the hatchery before they've even been dip netted, and then as they're ejected into their new home, and see what is the difference, and see if we can figure out from the STP-chip data what is the stressor that is causing the biggest change in their gene expression profile. So the last example I want to give, again, is a proof of concept. And this work is in Mark Shrimpton's lab, and it's being headed by HQP Carolyn Napper. And this is in partnership with the Gitnyao First Nations. And they're interested or concerned about environmental stressors impacting the sockeye salmon that they have a traditional fishery for in terms of decline, and increased numbers of resident sockeye salmon, which aren't as valuable for food. So residency in sockeye salmon, so quality, numbers, and residency. Pre-1950 escapement of the Kitwanga sockeye salmon exceeded 10,000. And it was a key food fish for the Gitnyao people. But according to Mark Shrimpton, the escapement in 2019 was 125 fish. So this is dramatic decline. And there are lots of possible stressors that might be associated with that. But on top of this is the large anadromous spawners, which are preferred, are decreasing, but the number of small green spawners, you can see in this picture, are increasing, and those are the putative residuals or non-migratory sockeye. So the design or the question for this project was, what are the physiological consequences of rearing in the lake for the juvenile? Is it the changes in the lake that's leading to the decline of the migratory and increase of the resident sockeye? And the approach that the Shrimpton Lab's taking is to look at smoke quality in terms of classic gill sodium potassium ATPase activity, but also using the osmoregulatory stress response transcriptional chip. Part 2 is lake sampling in the spring, summer, and fall to assess the response of these fish, these developing fish in the lake that are associated with high temperature crushes at specific times, not only high temperature, but low DO. And so using the STP-chip will allow Mark and the Gitnyao people to determine what are the critical stressors and can we reverse or mitigate any of them to increase the available food. Okay, so this is the scorecard. We're doing single controlled stressor trials. We're doing multiple controlled stressors, field gradient, mesocosm, and partner proof of concept. And you can see we're on target for proof of concept and stressor, single stressors, but we're behind on the multiple control stressor and gradient mesocosms. So if you're thinking of starting a new project, think about this. And then I think we have time for some questions. Yes, we do, about 10 minutes. And so questions for both Ken and I, depending upon which type of question. Tina?
I was just going to say, I really like your explanation of what a chip was. I think that's really, really helpful. And I know when we're calling it the STP chip, when I work with Céline, Odette, it's called une puce, so that's a thumb. So there's a lot of translational, there's nothing consistent even across different languages. So I'm just wondering, is a chip the best word to use? Because we've also run into that problem where people think it's like a microchip that you can actually just inject it in the animal, and then you can just put like a reader over it, like a PIT tag, and then it comes out with the information it needs. So should we rebrand, is what I'm saying.
I personally vote for doodad. It's
I'll take a note.
Tina, the term came basically from sort of the slang that Thermo Fisher used for their chips. It is fairly non-specific, and I don't know, Ken, can you think of something better than doodad?
I have a tendency to use thingy quite a bit in science, but yeah, I without going super technical with open arrays, it'd be something to think about, discuss further.
And there's some good comments. Sorry, Daniel. Oliver says, what about asking end users? Steve thinks it's a good call, and we could definitely bring this up to the end user committee for their input and reflection. That was very, very good.
And it would be unfortunate if our use of a term ends up being a barrier to adoption. Thanks, Tina. Amy?
Not a question, but I just wanted to make a comment about the potential of the non-lethal sampling. And I think that's really interesting because in some of the focus groups, especially with members of the public, the lethal sampling, even though it would be less than traditional methods, was still flagged as a potential concern.
Yeah, that's an interesting point, because our end users identified the non-lethal or even the non-invasive, the environmental RNA, mostly because they didn't want to hurt the fish they're trying to preserve. But public perception, that's really important. Thanks.
Nick?
Yeah, Daniel, I was curious to hear more about the saprinid chip and whether there are any plans. The validation of the salmonid chip was a good place to start because there's relatively few species and a lot of sequences. Now when the saprinid 600 species, persiform a lot more. I may have an opportunity to get my hands on a variety of different saprinid species, so I'm wondering where that's at.
So now I'm going to do something mean. Patricia, can you say anything about where the saprinid chip actually is?
Sure. Yeah, I'm still in sort of the beginning design process of the cypriniforms chip. So what that looks like is I've been taking the reference white sucker transcriptome produced by the Jeffries lab and searching for the target genes and then aligning to other related species within the order.
Maybe Patricia can reach out to you on that, Nicola, as well.
Yeah, there are colleagues of mine who are going to be doing a survey of saprinids in southern Ontario, a large number of them, and collecting them for another purpose. But it seems like there might be an opportunity there to do some work.
This brings up, Celine's not here, but I'm going to mention it. We have all sorts of RNA-seq money. So maybe we could grab some samples from those saprinids and get the transcriptomes done.
Okay.
Good. Still time for a couple more questions. I don't see any. So let's move on to Tina. Do you want to do activity 5?
Perfect. Okay. Well, we're hitting the home stretch, people. Thank you. I hope you've been eating.
Hey,
So
Tina,
you stay energized for the next 15. Yes.
we've got
Go
a
ahead.
blank screen, or I do.
Oh, again? Okay. Okay, hold on. I'll try this again. Okay.
It's because we're forcing you to
see.
use Teams.
No, that's okay. Is this working?
Nope.
Oh, still a black screen.
Ah, we work.
Yes. And you've
Okay.
also disappeared too, whether that was intentional or not.
All righty.
You're back.
Well, good thing is Paige has me on backup. So let's see. I'm going to turn off the camera, turn it back on. I'm going to stop sharing. This is going to be interesting. Okay.
Yay.
There we go.
Perfect.
Really?
Yep,
Yeah.
you're good.
Okay.
Well, you'll have to say
okay,
next
but you're
slide.
not seeing. Okay, perfect. I love this.
We can't see you,
But
but
let
we
me
see
just
your
try
slides.
just in case. Yeah, okay, but let me just try one more time. Just one more time. Just to make sure. Nothing? You don't see anything?
Nothing. Neither
No,
you
and you
nor
did,
your slides.
yeah, you disappear as well. And ironically, on my screen, it's actually not black, it's gray. So, I mean, we're getting lots of different varieties of not working.
Okay. Well, please, Paige, can you put it up for us? Thank you. Okay, awesome then. So y'all probably all know me. If you don't, I'm Tina Semenuk, University of Windsor. I am the person in charge of my wonderful colleagues in Activity 5, which is all about gels. And basically the triple E stands for ethical, environmental, and economic, as well as the legal and social aspects of our genomics tool. So this activity involves around 6 main PIs across 4 universities, obviously along with their students, and it really comes down to the commercialization of our genomics tools. Next slide, please. So the first, just to set everyone again, we've got 3 toolkits in GEN-FISH. These toolkits are the fish health, and that's your STP-chip. It's the fish survey toolkit, and that's the eDNA. But then we also have the decision-making toolkit, and this is what everything narrows down to. And this decision-making toolkit, the premise is to be able to assess our genomic toolkits, whether or not it's going to be acceptable, because at the end of the day, even though our assays may or may not work, they're working. Daniel, you can now breathe because Shaheen said that there's going to be some really good, eRNA. But still, even if it works, is it going to be adopted? And so the whole point is to work collectively in the economics and the ethical and the legal and the environmental realm to see whether or not there are any barriers to potential adoption. Next slide, please. So the first colleague that we have, and they're actually here with the exception of Tongji Lee, is Dr. Amy Fitzgerald from the University of Windsor. And what Amy is studying along with her HQP are the ethical concerns using these tools. And that's because really going over the ownership and privacy of eDNA, genetic information, transcriptomic transformation of the nature of life. And so it's really, really important to be able to get a handle on the ethics behind our tools. And the methodology that Amy engages in is literature reviews, media sweeps, case studies, and focus groups. Next slide, please. Along with Jennifer Halliday, her PhD student, and in my more recent up-to-date presentation, I had the student's photo, so my apologies to everyone. But this is an example of a media sweep. Media tends to do 2 things. Sometimes it reflects what society is feeling, but it also drives what society should be feeling and what the concern should be. So what Amy and her team does is a media sweep from the last 20 years or so, and looking in this case, Amy's already done it for eDNA, and I presented that at our last year's AGM. And this time was to look at transcriptomics and how it's being presented in the media, and in two large publication types, one the New York Times, and the second one is the Globe and Mail. So over the past 20 years, how is transcriptomics being mentioned? How is it being mentioned, for example, gene expression, so keywords, but going above and beyond GMOs, genetically modified organisms. That's usually what it's typically framed in. But Amy was going a step beyond that to take a look at the socio-ethical implications that are beyond just simple GMOs. And so there are about 9 or 10 sub-themes that emerge, and you might be able to see it across your screen, from legal to mental health, privacy, ownership. And so these are when, themes in which transcriptomics has been used. But when we're taking a look at the socio-ethical implications, it's really only the nature sub-theme that contain that kind of discussion. It was done so framed in terms of what we consider human nature. And so this is what the title of the article is that's been submitted to Public Understanding of Science, and that's the molecularization of life, which means we're growing our capacity to be able to control, manage, engineer, reshape, even modulate the very vital capacities of human beings as living creatures. And so that's how it's being framed. So it's really, really interesting information, and it just goes to show you where the media is right now. And there hasn't really been a discussion yet into the environment. It's still really human-centric. Next slide, please. But Amy's also focusing on doing focus groups and one-on-ones. Her group has completed about 14 focus groups and one-to-one interviews to date. And we've heard some emergent themes Amy's mentioned already, but here's some other ones that have been touched upon that have emerged from the focus groups themselves. And so can I just have the next 2 slides, because it's our animations? Thank you. So the first one is, okay, is there going to be informed consent when we're collecting DNA? And so again, this is transcriptomics and eDNA. And so there's this concern about human DNA being collected. And then, okay, with the DNA and the samples being collected, will there be any security over the and storage in these reference samples? There's also, interestingly enough, concerns raised about decision errors, the unknown that's going to happen, whether or not commercial impacts are going to exceed the environmental interests, so putting ahead economics and profits versus environmental impacts. And then again, there's a social connotation emerging from these toolkits, these genomic toolkits, and those could be potentially negative impacts on already disadvantaged social communities. And again, there's another kind of emergent theme is that the scale and scope of environmental degradation does require novel approaches. So it's the other side now. It's saying, okay, here are concerns, but then again, here's some hope and here's what we think can happen. So I know that Amy could discuss more a little bit about who she's actually targeted, but I do know it's some fisheries managers, potentially some public. We did one with Amy last year at our previous AGM, and so Amy can get more into the details to give you more information. Next slide, please. Perfect. So this should lead us into activity 5.2. The lead collaborator on this is Dr. Tongji Li from the University of Guelph, and she's studying, again, the social, economic, and regulatory factors that can impact the acceptance of and adoption of these genomic toolkits. Tongji doesn't just focus on the immediate end users, but interestingly enough, to look at social, general public acceptance. And the reason for that is because sometimes a lot of industry may not want to adopt a technology if there's any kind of negative connotations in terms of in the social realm itself. So it's also not just the targeted end users, but general public acceptance. And what Amy does, she uses behavioral economics. She uses behavioral experiments to try to figure out, are there any kind of behavioral nudges and economic incentives that could influence not only the adoption, but the rate of technological diffusion. And so how quickly something can be adopted. So I've talked about some of Amy's projects in the past, but next slide, please. And I'll be talking specifically about a type of behavioral economics. So next slide. Perfect, thank you very much. So this is work done with a student, Kane Boyle. And I just want to read you out this statement. So bear with me, I know it's a lot of text on the screen, but just listen to the keywords. If eDNA monitoring technology was adopted in a commercial fishery, and that led to improved estimates on the maximum sustainable yield, how would that affect the behavior of the fishers just using that fishery in terms of overfishing and conservation? A couple key terms here, and we don't necessarily want to rely on the industry itself, but what we can do is we can broaden this out, be more wide about this without targeting a certain group. And so in terms of a behavioral economics experiment, we're going to take this and we're going to target a broader, let's say the public. And next slide, please. We're going to ask the following question. How does the exogenous reduction in threshold uncertainty in a common pool resource change the appropriation and conservation of that resource? Okay, I've underlined these words because they've just been substituted. That slide before, I just said the exact same thing, but I was very specific. Here we're taking generalities. The exogenous reduction in threshold uncertainty. So here we're using a tool to help us maximize our maximum sustainable yield by reducing the unknowns about that problem. But yet this is a fisheries. This is a commercial fisheries, which happens to be a common pool resource. And so does this reduction in uncertainty affect a common pool resource and how we exploit it? And so you see here what Tongji and King are doing is they're setting up an actual common pool resource game where we've got multiple people competing against one another in a common pool resource, again with money. And what's actually being manipulated here is the uncertainty and complexity of the common pool resource, the uncertainty surrounding that. And this is where the technology comes in. This is where our genomic tools can be substituted in. And it basically gives us insight into that reduction in uncertainty. Does it lead to more sustainable use? And again, we can change the industry. We could look at recreational fishing. We could look at community fishing, subsistence fishing. So there's a really wide general applicability. This project has just started, and I'm really excited potentially for Kane or other HQP to present this in a lot more detail in our next AGM. Next slide, please. So we have our third objective in activity 5, and that's the ex ante evaluation of innovative technologies. Ex ante is just a nice fancy word for saying forward-looking assessment, forward assessment. And of course, this is where everything comes together. And this is being led by Dr. John Levernois from the University of Guelph. And the whole point here is to really look across all of our activities, take all that information together for two main purposes. The one, obviously, is to create this multi-criteria decision-making tool. Peter Martin talked about it yesterday, gave us a really nice presentation. But the second part, and I just want to stress that again, I kind of mentioned it in our last workshop of the day, and it's really to develop a translation plan. A translation plan that can consist of possible mitigation strategies for implementation. So yes, we can say, okay, you can actually, it looks like through this multi-decision criteria tool, that eDNA might be the right path forward. Okay, well, then how do we get there? What's all the extra information that we need? Next slide, please. So John and his crew have been doing a great job of connecting with our leads and other members in activities 1 and 2, and again, eventually in 3 and 4, and a little bit of progress there as well, to obtain data, interpret field recorded data, actually curate it all. I think there's a nice Python workflow because there's been so much information that we're gleaning. And this is actually really, really important information to be able to start collating a lot of this intensive computation. Next slide, please. And all of this information is being fed into this multi-criteria decision-making model. Peter Martin gave an amazing talk yesterday about everything that it is. So I'm not going to go into the detail again. I think it was a really, really good overview. But what it's doing, it's going to be incorporating simple user input, the preferences that users need and what they require, project requirements in terms of the site, sampling characteristics. All of those site characteristics will go into these decision-making models to say, okay, are these genomic toolkits the right tool for the job? And so the hope is to make this an interactive online tool as well, where we're going to be publishing it. Again, I think there's some questions about in terms of the eDNA folk, where all the data are going to be, but this is something that's going to be interactive. It could be GitHub. It depends on how complicated it gets behind the scenes, but it should be a pretty simple, hopefully, user face that goes along with that. And so thank you to Peter and John for working on this. Next slide, please. I would be remiss in actually not also mentioning Doctors Stephen Cook and Vivian Guyen, both from Carleton University, with their HQP, Andrew Haworth, because we step outside the strictures of what Activity 5 mandate is to show that we're doing a lot of co-production work as well. We've already seen co-production work through Margaret's and Nick and Ken and Daniel and Matthew and Shahin. So working with our end users is great, but we're also doing it in Activity 5 to help out to identify these barriers to adoption. And so I just wanted to show you some of Andrew's work. And I know he presented yesterday, but there are a couple of things I wanted just to bring to our attention in terms of the work that Andrew's done with Vivian and with Steve. So next slide, please. So the first one is, I think I just gave a brief overview last year, but it's come out this year, this paper by Andrew, in which they conducted 50 interviews of fisheries managers across the country and basically wanted to look at the perspectives of these practitioners regarding the state of the fisheries management and how they feel about new technology, including genomics. The largest take-home message was everyone's really strapped. Like social capacity, financial capacity, and people are kind of running just a lot of lack of time and person power to be able to do what is needed to be done. And so there's a really, really good place for genomics to come in because it's responsive, it's immediate, it's legally defensible in certain cases, and that's what we're striving towards. We're encountering novel problems, and so this really has the potential to forward some novel solutions. And the fact that it can be so efficient, and I think that's the key, it's the efficiency in terms of time, people, and resources, so that less at the end of the day is more. And this is a feeling amongst all fisheries practitioners. And I want to give a huge congratulations, and everyone join me because this paper was selected as the Editor's Choice selection for the September 2023 issue in the Canadian Journal of Fisheries and Aquatic Sciences. So well done, Andrew. This is amazing, fantastic. But I want to do a follow-up on Andrew's work. So next slide, please. And this is basically almost like a continuation, but instead of focusing on fisheries practitioners, we're now looking at the supporting cast. And when I say we, I don't mean me. It's no royal we. I actually mean Andrew, Vivian, and Steve. But what they're looking at is really who are the supporting cast members across Canada. This is a beautiful photo, and it just shows the node, how everything's connected by from the different practitioners, from federal government, municipal lower government, to Indigenous communities, academic, NGOs. And basically, are they involved as a central role or supportive role in looking at fish, habitat, people, or other? But I want to just focus and draw out the supporting cast, for example, NGOs. And I think this is really important. We don't really a lot of the discussions we've been having yesterday and today, we're not looking potentially at our other users. And I think this is a really good insight into that. And when asked about, and I asked Andrew, like, what do you think the acceptance is for the supporting cast in terms of adoption of these toolkits? And the one was that, okay, right now, for example, I'm just going to use NGOs. They're used more as a consulting, right? As a consulting role, they're picking up the slack of, let's say, government, and as a consequence, they're not always drawn on, but they kind of fill in the gaps and they kind of help stick their finger in the dam of all the work that needs to be done. So there's going to be really strong acceptance of this fish survey toolkit, because again, as teams are having and struggling to do their basic monitoring, NGOs can fill that gap nicely with this tool. The health toolkit, there's this impression that it might be a little bit more difficult to adopt. If these NGOs are doing it themselves with a really strong, let's say, specialized question in this research and health, then I think it could be adopted a little bit more readily. Or if the team that they're working with, that's their focus. It's not that there's this impression that it's not that fisheries managers devalue being able to look for biomarkers of impending stress, sublethal stressors. And we saw that in terms of the work done on the CT max, and okay, is it 18 degrees, we could start to pick up these signals. That's really important, but everyone is still really trying to figure out how many, how big, who questions. And so NGOs themselves are potentially less likely. Again, this is without looking into further, but we're starting to signal that there will be heterogeneity and different typologies across our end users that really need looking into. We just want to make sure everyone's voices are represented. Next slide, please. And I also want to highlight now in that we step outside activity 5 and we're crossing the different activities. And you can see here this paper that was published in 2023. You might recognize some names of these co-authors in the Canadian Journal of Fisheries and Aquatic Sciences. Dr. Thais Bernos was a postdoc, I think was a PhD, then postdoc in Dr. Mandrak's lab. Nick, you can correct me in the chat. But the co-authors put out this really great paper to look at Canada specifically. What are some of the challenges that Canadians face in terms of adopting eDNA technology? And you can see we have some end users on our, I think our panel here as well. And I think we had a discussion yesterday that although we think Canada is leading the charge, it's actually Australia and New Zealand that are using the eDNA technology primarily and much better than Canada is. And some of the reasons why, maybe because Canada's broad, we're heterogeneous geography, we've got specificity of place and location, we've got areas aren't all connected. And so, you know, how do you reach the, and Matthew, Dr. Yates, you basically said up north, these are locations that are hard to get to. So again, that's one of the barriers. Another one is our complex political landscape. Daniel had a joke at the end about, you know, climate change, changing government, it doesn't exist anymore, poof. And that's something that we're facing as well in terms of how much support this is getting. And again, we've got the cultural diversity of Canada. We can't forget that we're working in unceded traditional territories of First Nations, Inuit, and Métis peoples, and that, well, first and foremost, the permission and the co-production with our primary land users, rights holders, and local knowledge holders should be at the forefront too as we're embarking on this research. That's all I have. Thank you for sticking with me 3 minutes past for the technical difficulties, and I'm happy to either take questions now or take questions in our question period because we all need a bit of a health break right now. So thanks, everybody.
Thanks, Tina. I think that while it's fresh in our minds, let's see if there are a couple of questions, and then we'll just delay the startup for the general discussion. So any questions for Tina?
I'm just reading the chat.
So I don't see any. Tina, I have a question for you, and you may want to bring in other members of Activity 5 who are on the meeting. Yesterday, we were talking about adoption, and for the STP-chip, it came out that there are certain audiences that we figure are going to glom onto that, as opposed to the eDNA, which is sort of broader. So do you think we have to worry about adoption at the broader context or a more narrow? Do you think that's an issue, or can we just say it's okay, the fish hatchery managers love the STP-chip, we're good?
I mean, to give a flippant response, it depends on how much money people want to make out of this. So, you know, broad is good. But in terms of the utility, I don't know, Steve, Andrew, and Amy, if you brought in, what were the kinds of the comments? Because we heard yesterday that government is on board. Feds, they like it. In terms of the STP, eDNA, maybe not. In terms of other domains. But what are some of, like, what are your impressions? So Andrew, you've spoken to a lot of different fishery managers, you've spoken to NGOs, you've gotten feedback, one-on-one quotes. What's your take?
I think anyone who has money to spend and has sort of an acute issue that the STP chip can help with. The aquaculture example, I think, is a perfect one where they're interested in osmoregulation and growing fish fast and making money. That's the kind of people that I, just from my limited experience, would expect to adopt the STP chip. Folks doing more routine applied work in government, less likely, I think, but more likely to have use for eDNA if it saves them time and money and it frees them up to do the things they want to do.
Yeah.
Maybe it's also just thinking that, you know, maybe we're thinking about this the wrong way. Maybe practitioners, yeah, I guess one question is how often are our frontline practitioners going in the field these days. I know I don't know if Chris Wilson's still on board, but the local resource management staff in my zone, fisheries management zone, aren't allowed out of the office. It's the conservation officers that are on the road. And they end up relying on their science staff to support them. And so, in other words, should we really be trying to build capacity and sell these tools to the science staff, the stock assessment staff, and so on? In other words, almost working at the headquarters level for the stock assessment side of things to get sort of institutional buy-in and then work that in. And then, of course, the research scientists who are the ones responding to the resource manager saying, wow, I'm getting a lot of phone calls about this issue in this lake. What should we do about it? Think about the resource science, the research scientist being the one that would likely pick up and run with this, and not expecting the frontline resource manager to be able to keep up with all of this stuff. Anyways, it's just a thought. Maybe for the resource management, it's just they're too busy, neck deep, trying to keep actors happy.
Amy.
That summarizes it pretty well, although it is getting better.
Based on the focus groups, I've noticed a difference in that the recreational fishers, the public, the environmentalists have questions about eDNA and the security of it. Is human DNA being kept? What are the potential implications there? But the researchers, some of them have voiced concern about the implications of the health chip, the STP-chip. Like what will be done with it if, commercialization and possible manipulation of fish down the road and then potentially released into freshwater. And so those, whereas the public doesn't, and recreational fishers really don't problematize that side of it as much, but the researchers seem to.
That's really interesting. Daniel, does that answer your question then, in terms of targeting?
I think it may come back to the question of education. And it may be that the genomics part is just a lot tougher to understand. And so it's more something I'll worry about later. It's not like something in my backyard now. And I think that if the education component might be critical, but it might actually raise barriers, which would be pretty ironic.
Mm-hmm.
And then you start to think about, if you've done an undergrad degree in resource management or biology or environmental science, how much of this did you have? And if you went to school 30 years ago, are you likely to be trained or retrained on how to do this at a so, and again, it goes back to research scientists actually have the time and mandate to keep up with emerging, evolving technique, especially if they're kind of in their wheelhouse. So yeah, and even you think about the workshops. This is pretty sciencey stuff. These are the kinds of workshops that could would normally be put on as maybe professional development at an AFS conference. Maybe we could do that kind of thing at an SCAS conference. But those folks aren't coming to those venues. They're not allowed out of their district, or province or territory. So yeah.
Oliver?
Yeah, this is a little bit outside the scope of the current questions, but I remember when we were first getting together to plan sort of GEN-FISH and what we were going to do for experiments. I remember early on, we were wondering whether we should actually try to pitch using CRISPR and gene editing to basically manipulate fish and release them. And we thought, okay, you better not go that far. It's way too early for that. But you know that the FDA just approved the first gene editing, right, for sickle cell anemia just a couple days ago. And so it's already going to be in the public's eye pretty soon. You know, pretty soon we're going to have sort of GMO humans walking around who are being gene edited, for instance, to solve a disease. I'm just curious about what everybody thinks in terms of whether things like CRISPR and gene editing are making any of these discussions easier or harder. I mean, Amy, you mentioned that, you know, researchers are worried about commercial producers running away with things like this. Pretty soon, I mean, we're already tripletizing commercial fish in Canada for production and eating. I'm just curious about whether things like gene editing are going to make these discussions easier, harder, for instance. And I'm curious about whether whoever's going to take on GEN-FISH Mark II after this one's done, whether we actually feel comfortable sort of going in and actually proposing things like CRISPR, because by then, if we don't, we're kind of going to be behind the times already for what sort of everyone else is doing sort of health-wise and commercially. I was just curious about what people thought. Or not.
You just can't, it won't be marketed as a GMO.
Yeah.
I would say, like, when we were talking about it, like, Chris Wilson's here. Anyone who works in the hatcheries, we're doing selective broodstock. We're not doing direct gene manipulation, but we're doing it indirectly all the time through our selective practices. I think it comes down to what Daniel said. It's just education, and these are buzzwords and keywords, right? When we're not going to call it CRISPR, but we can call it, for example, sickle cell anemia, not mRNA vaccination, apparently, but we have to try to find words that don't upset people. And it comes down to, I think, focus groups again. Sorry, Chris, I evoked you. Would you like to comment?
Yeah, that had to provoke a response. Sorry for the cat. Yeah, I think there was very good reason that it needed to come out of the first round of the proposal. I mean, now that could go forward for a follow-up one. There will be lots of concerns in terms of selective breeding of hatchery fish. They try to avoid that. Now for aquaculture, that's well advanced, and there's definite uses and benefits for that. There are a lot of public concerns for genetic rescue projects. I have been accused of making Frankenfish. And then when it worked, people were quite happy, which were not Frankenfish, but basically the gene editing, the public perception from what I have seen, there's still GMOs. Wrongly, I think. I think if there is a follow-up one, there is room for this, and it can really build on the the gene chip transcription data in particular. There could be a good case for this, but it belongs in a follow-up. I am biased. I think it's very good that it did come out of, was removed from this first generation one.
Yeah. Well, Joe says he's going to kick us out because he's got to end the meeting for the, how long we could record for.
No, I'm just going to close it when everybody's
Joe,
done.
I'm trying to get a break here. I am trying to end.
Don't make me the bad guy.
Bathroom.
Don't make
me the bad guy. Okay, so Tina's saying that we're going to end and we're going to come back at 2 o'clock. So how does that sound? James, the last words.
So let's come back at 2:15. So we still have a 45-minute break to go for lunch. So we'll reconvene at 2:15 and take it from there.
Super. Okay, thanks, everybody.
Thanks. I guess we'll get started. And I think that the approach is to talk about moving forward with each of the toolkits. And if we can highlight specific action items or goals for the last 18 months of GEN-FISH, that would be good. So Margaret and Matthew, you have the floor.
Yeah, hadn't thought that far ahead for this, but yeah, I mean, action item for activity 1 for the remaining 18 months. I mean, very definitely finishing off the assay design and validation, you know, objective 1.1. I mean, that's really, I think, yeah, the bread and butter of activity 1, making sure that those assays are developed and validated and ready to share with the general public, with end users. Yeah, and with all of the documentation that goes along with it to make sure that they're truly useful.
Well, Nick, I just noticed that Nick is back. I don't know if Nick, you want to handle this one, or do you want me to respond?
Well, I'll take a cut at it. And Matthew, thanks so much for covering for me this morning. I caught the tail end of your presentation. I really appreciate it, and I'm sorry I couldn't be here yesterday. Well, you know, we have two main things going on in activity 2. One is the 500 water body survey. I think the key there is we need to get the results so we can hand the database over to the database team that's going to look at, let's say, emergent properties related to what may influence detection. So that we need to get that database as soon as possible so that Sapna and Pedro can start working on it. And then, you know, we have a lot of case studies. They're in various stages of completion. I mean, I think the ones that have they all look in good shape to be completed over the next year or so. And what I foresee on that front is almost like a bookend to the Thais Bernos paper, which talked about the challenges, but give examples of how we could address those challenges through review of some of the case studies. And I expect all the case study authors to publish their own work, but I will invite all of them to participate in a synthetic paper that again, I hope we can use to show that how the potential of the methods, as well as the potential to address some of the challenges that were identified in the initial Bernos paper. And that's how I see it. That's what I see. That's how I see it playing out in the next year or so. We do have to still get over that backlog of samples in the lab, and maybe this is where I turn it over to Matthew, so we can talk a little bit about that.
Well, we're working on that. We did get a whole bunch extracted this summer, so there's like 30 plus plates of samples basically sitting in freezers in Guir, and we're starting the process of metabarcoding, and hopefully soon we'll be starting the process of applying the open array chips to the samples as well. So that's ongoing, slow, but still going.
John?
Yeah, could I ask maybe a question that's kind of general, at least initially, but it'll get more specific. So Margaret talked about validity. Matthew talked about ground truthing. Obviously, those are two very important pieces of information for anybody thinking about using eDNA instead of what they've been doing in the past. And obviously, it's going to be a key bit of information that feeds into the decision-making model in activity 5. So my question is about measure. How do you measurement? How do you measure, or have you thought about a metric of truthfulness or validity or accuracy or reliability? It's unidimensional or multidimensional, you know, some kind of metric that would conveniently, like the results of your testing for validity and truthfulness, ground truthfulness, a metric that could fit into a decision-making toolkit for the for the end user to help decide whether or not to go ahead and use that one. We're trading off cost and reliability and other things, but we've got to have that kind of metric. So it's just a question about is there thinking around producing metrics that could be useful in that way? I see, and Matthew's got an answer, I see.
Yeah, well, I mean, so there's a couple of ways you can kind of, from a like ground truthing perspective, approach this problem with eDNA. And I mean, this is something that's been explored in a lot of studies. I expect that we'll probably just do it at a larger scale once we have the final data set produced. And Sapna and Pedro will be a big part of that. But I mean, a basic metric is just the probability of detection based off of sampling effort. And so there's a large number of case studies that have, there's kind of two ways I think that it's envisioned that the final activity to water body will sort of approach this question. The first will be, there will just be a large number of case studies, somewhere on the order of 1,500 or so of the samples we've collected have paired traditional assessments of species present in an ecosystem. Just projects like Ian's stormwater pond, the agricultural drains ponds, a lot of the work coming out of the Shrimpton lab. There's actually just straight up temporally paired comparisons with eDNA versus traditional methods. And so you can look at, what's the relative probability of detection for like per unit effort of detection, either eDNA samples or net sets or what have you, electrofishing effort. And that's a relatively straightforward metric. You have like, okay, one method works better in this context, another works in this context. You might be able to get even finer scale inferences based off of like environmental characteristics, depending on what environmental data has been collected. Like Dr. Docker mentioned in the roundtable discussion, things like pH could affect the reliability of eDNA or temperature, things, environmental parameters like that. And the other way you could kind of think about this, and I don't know as much about this because this is kind of outside of my realm of expertise, and I think this is what Sapna and Pedro were planning on doing, is basically looking at historical occurrence data and sort of modeling like, okay, we know these species should be here based off of the environmental characteristics. Is eDNA detecting the communities that we would expect to be here based off of, this historical database? So that's the way I think that folks are kind of envisioning validating or ground truthing the eDNA, at least relative to other more traditional methods. And there's other ways you can even crack the question or take a look at this question too, because you can look at things like quantitative estimates of like how closely do these parameters relate to abundance, how close, if you're looking at eDNA concentrations, how much do you need to sample an environment in order to get an accurate estimate? So like you can do power analyses and these types of things. So those are a lot more technical and really specific, because those types of questions are very like case study specific, because it's going to be different for every species, like how they respond to the environment and the eDNA that they produce. There might be general trends, but I got to imagine that it's going to be pretty contextually specific for those types of questions. But yeah, on the broader scale, it's going to be the paired comparisons and then the historical database thingy.
Chris, do you have something to add to that?
Yeah, two things actually. One lab-based, one field-based, and still in answer to John's question. So I think one thing Matthew didn't mention, because it's sort of assumed at this point of going through the process, this came up in earlier presentations, but the importance of validating your detection ability with a mock community. So that if there's any sort of amplification bias or detection bias when you're looking for multiple species, that's why it's so critical to have that front end. So for instance, most of the so-called universal fish metabar coding assays don't work well for cartilage-based fishes. So if you have a negative bias, that's detected in the mock communities, and you can correct for that, either design a new assay, which is a lot of work, or just calibrate and say, if we're only 50% effective of detecting species X, then let's just double what it tells us. So that is the lab-based one. The data are in for that already based on all the work that's been done on testing the assays and using the mock communities. The other one is field-based. And Nick and Margaret both alluded to this earlier when was talking about deep water sculpin. If you want confidence for detecting something or being able to say if it's there, the sampling design should reflect that. And this will also encourage buy-in from the field practitioners. So if you're looking for a benthic species, the sampling design should reflect that. You go to where the species are. Now, with mixing, you should get detections regardless, but if you want a more accurate or nuanced detection, the sampling should reflect the question being asked and the species ecology. So hope that helps.
Nick.
Yeah, just to follow up on Chris's comment about the field work, and you know, Matthew mentioned detection probability. To me, this is the key next step with a lot of these data that have been collected, particularly in paired samples, where we would determine the detection probability of eDNA versus the conventional sampling. And in fact, Ruchira Castaneda has published some on this already, some papers on this already. And it doesn't matter if it's eDNA or any other method. It's just compares. It's the straight idea of comparing detection probability across methods. And I think this is, you know, this is not necessarily going to be done in the next year, but I expect to be mining these data for some time to come, because I think this is where we have to go. We have to determine the detection probabilities, and then the second part, and Matthew alluded it, and Chris both alluded to it, was how, you know, how many samples do we have to take, you know, in order to detect? And, you know, so for example, DFO is interested in goldfish and ponds. And in fact, this is a question that was coming up earlier. We're helping DFO do some analyses where we are looking at the characteristics of 100 ponds and seeing if we can predict which ponds would have goldfish in them based on the characteristics of the pond, whether or not they're in an urban or rural environment, you know, all these different characters. But we actually need to know whether or not goldfish are in those ponds, so we can use that as the dependent variable. And we can't do it yet, but I already asked Matthew and Ian Smith to give me an idea. You know, how many times do we have to go and, you know, how many samples do we have to take from that pond? Can we go to the pond and just take one sample, and then we know whether or not goldfish are there? Or if not, how many samples do we have to take, and where do we take them from? Right? To me, these are the key questions that management agencies will want to know. And one of our early case studies was where we paired eDNA sampling with broad scale monitoring. So we used exactly the same stratified random method that M&R uses to put nets in the lake. We did exactly the same thing with eDNA. Now, you know, once we get the results, we have to go back and determine, did we oversample? Did we undersample? To me, this is where we should is the next step, and then layer on top of that the abundance question: how much sampling and where do you sample if you want to determine abundance as well. So I that's where we should be going. I just don't think we'll be there in the next year. I'm concerned we may not even be there in the next year with the database analysis, because we do need the database in order to actually do that work.
Thanks, Nick. So I would say, from my point of view, that in fact, John, the reliability or efficacy score for eDNA will actually be a function. And as you get higher efficacy, the cost will go up. Because the bottom line is, if you're willing to do 100,000 eDNA samples on a lake, you're going to have very high detection probabilities. If you do one, it's going to start getting iffy. Tina.
So what I'm thinking is that we need to come up with a list of data knowledge gaps with, I think with the overall theme of how is it going to feed into our decision-making toolkit. And so where we might not have a definitive answer for you, John, it'll be a slider. So in terms of where the practitioner can be, do they want to have a high sampling effort with associated cost, or a low sampling effort, but then the probability of detection is much lower? So they'd have to decide themselves along that slider where they want to fit in. So we might not be able to give definitive answers. but we need to come up with the questions though. And so how can we work collectively to come up with the questions that we think our end users will need to know? Go to our end users, obviously, but I want to start brainstorming in terms of where can we go to at least questions are guessing that we actually, obviously we're experienced, we have our connections, but we don't necessarily want to just take it from us, but from the end users themselves. So what are the mechanisms by which we can tap into this information? We've got fisheries management, we've got the workshops coming up for the GLFC science transfer project, for the STP-chip, or the omics, I'm sorry, for the omics. But what are other avenues we can pursue? So any ideas?
So, Tina, you're talking about specific ideas of how to approach this?
Yeah, because I just don't want to talk again, like we need to do this, we need to do that. Yes, we do need to do this. How are we going to do it? I like action items going forward. So we're going to put together a workshop in February, and here's who we're going to invite. We're going to have an end user meeting in April, and we're going to invite these folks, and we're going to directly ask them specifically these questions. Those are the type of action items I'm thinking of.
Yep. No, I think that we're reaching the point where those kind of actions are going to become critical. And I think that's up to the Science Management Committee to actually move forward on. But I think it's good. That's a placeholder for that. The other thing is I think maybe activities 1 and 2 might want to consider going to monthly brainstorming meetings, as we've decided to do for activity 3 and 4, to identify issues and try to solve them. John, you're next.
Oh, thanks, Daniel. So I'm just trying to make sure I understand what's been said. So Daniel, I like I like what you said about there being a function that relates probability of detection to sample size, or how much sampling effort, let's say. And Tina's version of that, which is a slider, maybe a little less sophisticated than a function. But based on what Nick said, we're not there yet in terms of having the information to even produce a slider, or definitely not a function. But if we had that, I can see that would fit nicely into a decision-making model in which the choice variable for the user would be what detection probability do you want? And then the function or slider would spit out, okay, this is how much sampling you're going to have to do, and then this is how much it's going to cost. But it seems to me like we're not going to be there within the next year or so. So that's my first question. Am I understanding that correctly or not correctly? And if that's correct, then based on what Matthew said, we do have information that's not functions, but it does definitely we have sort of comparative information on probability of detection for given sample size of different species in perhaps different environmental conditions. And so, as an interim approach, we would have to use that kind of information in the decision-making model until we've got the function that Daniel was talking about. Am I getting that right? More or less?
The timing is the tough part. Hopefully the generation of the eDNA detection data will ramp up, and we'll be able to start passing data to SAPNA and Pedro. And then I think the analysis of the factors that affect detection, including the number of samples taken relative to lake size, would be relatively easy for them to do as a first pass. But Chris, do you want to add to this?
Sorry, just a couple of things. There are a number of papers in the literature now talking about effect of sampling effort on species detection, both individual or targeted and community approaches. I've actually been surprised at some of the numbers, but they're mostly based in Europe, and their lakes are different in both size and qualitatively than we're dealing with in Canada. I've just put one paper into the chat that I think is quite good. Combines both sampling effort and detection probability. But for one thing that Tina brought up for inviting people to the February workshop, I mentioned yesterday, Fisheries Notions has been making great strides in eDNA. It would be very good to include some people from their national working group. In Ontario, Ministry of Environment, Conservation and Parks is going through a lot of angst wrestling with eDNA for detecting species at risk and also documenting biodiversity in parks. So they would be another group to bring in, likewise Environment Canada. And you've probably all been contacted several times by environmental consulting companies asking about eDNA. Something that the upcoming CSA eDNA standard that's looking at sampling design, they're wanting to bring in consulting companies to be involved in this from the front end, to be able to say, are we asking the right questions? And can we make it relevant? That was also one of the successes behind the Australia effort. It was a mix of government research, well, led by academia, government research, the consulting industry, and the end users that are hiring them. So mining, forestry, hydro development. So I would just, who are appropriate people on those lists, and make sure that it's focused from step one. So that's all.
Thanks, Chris. Nick?
I think those are all really good points. I just wanted to point out, perhaps to John, there may not be one function, right? Because detection probability can be largely context dependent, right? Could vary even for the same species between different water bodies. And I'm not sure if that's, I'm hoping that SAPNA and Pedro will be looking at that, but I'm not sure if they're necessarily taking a detection probability approach. But we have so many projects in there, including our case studies, that we could start looking at how much variation there is once we get the data. And I'll point out that one of the larger case studies is the one on ecosystem health in the Saugeen Peninsula that we're doing with Saugeen Ojibwe Nation. And I do have a student who is doing detection probabilities on the conventional sampling with the idea that once they do that, then when the eDNA data are available, they just plug in those data and then we'll be able to do the comparisons, and then we can start getting some of those functions at least for that area. So it's not, I sort of may have seemed like I was quite pessimistic about the timeline on this earlier, but you know, we are thinking of ways to get this done, and I do have quite a few people in my lab do detection probability, and once we have data, I can shift people onto it as well. So, but again, it depends on the availability of the data. But I guess my main point was context dependence. There may not be one function that explains everything.
Or it may be multiple variables within the function.
Yeah, could be a complicated function,
yeah,
or many of them. Thanks, Nick. That's good. I did just one more thing. So we've been talking about detection probability. Do we need to think also about like type 1, type 2 errors? Are users likely to be interested in those sorts of things? And do we have, or are we getting that kind of data to estimate those things?
Yeah, well, you know, detection probability is essentially looking at false negatives, the false negative rate, right? So yeah, we're getting at that. The false positive, well, that's, I'll leave it to the lab people to talk about the false positives.
Yeah, both of those are critical in terms of reliability, and it's part of the sort of activity one plan is to go beyond simply designing the primers and throwing them out there, but also validating them. One thing I will note about activity 2, the 500 water body study, I was just contacted by a Swiss group, and they want to sample 1,200 lakes worldwide. And their intention for eDNA is not just fish and not just aquatic organisms, but all organisms living in the watershed. And they're aiming for May 22nd, Global Biodiversity Day. We're supposed to go out and sample for them. So I sure as heck hope we don't get scooped on the 500 water body project. Considering they're just getting water samples at this point, they've got a long way to go.
May I suggest that you filter it through your EGF so it all turns up Chinook?
Low blow, Tina. Low blow.
I'm sorry. I blame Matthew. He brought it.
Got that problem fixed, I think. So
Mm-hmm.
the eDNA room helps a lot.
Well, our early work with meta barcoding, we kept coming up with tuna, cows, pigs, chickens. Good. So, John, I think you brought up a really critical actionable item for activities 1 and 2. I think, Margaret, you were totally on point in terms of we've got to finish developing those markers. Because remember that meta barcoding is kind of an add-on that we are told to do to GEN-FISH. It's actually the qPCR assays, which will have a very different detection detection probability curve than meta barcoding.
Yeah, and it's tricky because, the metabar coding, a bit of an add-on, certainly the optimization of metabar coding, a bit of an add-on, the open array chips completely added on to the original proposal. The challenge is that even though activity 1, if we can deliver 2 really good, well-validated assays for each freshwater fish of Canada, we'll consider that a big success, except that activity 2 and activity 5 depend on us going further than that to be able to have at least some metric of efficiency or effectiveness and some measure of cost, and all of those things are all going to be it depends scenario. But at least when people say, well, how much does it cost to run an eDNA sample, the answer is always it depends, because it depends on the sampling design, and the sample design depends on the question and all that sort of stuff. But at least if we can have, under these conditions, this is what it costs, or with this sample effort for this species, this is what our detection probability is, at least as a starting point. And as Nick said, at least getting an idea with the data sets that we have, what sort of variability is there. If there's so much variability that extrapolating from one species or system to another is almost useless, then at least it's good to know if the variability is not too bad, then we can at least, for starters, extrapolate.
I agree, Margaret. I think that getting those assays done and have some sort of validation data or performance data be a huge step. And at this point, both activity 1 and 2 are moving forward together. Oliver.
Just really quickly, to make sure that, by the end of this project, you get a deliverable for activity 5, is there some halfway house where you can sort of choose 3 species? Choose an invasive goldfish, choose something on the opposite end of the spectrum, something really rare that people are looking for. Nick's always looking for northern mad toms or, sand darters or something like that. And then something in between where you sort of expect it to be seasonally abundant, like one of the salmonids coming up a river, or something that's commercially important, but at mid-level, something like yellow perch. And then you could sort of just have 2 extremes and something in the middle where you could at least provide end users with, okay, if you just want to know where there's goldfish here and there's 8 billion in this retention pond, just take 1 sample and you're good to go. And this is going to cost you X dollars, $1 sign. If you're looking for something like Mad Tom and you want to be sure, you're going to need at least X number of samples. You don't have to be directly on the mark, and it's going to cost you $5 signs or something like that. And then just something, if I was an end user, I would just generally want to know what's the time, general amounts of money, and I wouldn't expect the team to be right on the mark just yet. I just want a general idea and then use that as a basis to add in more detail as more data comes in for more context and different water bodies and things like that. That way you get a deliverable, but you don't have to feel as if you're making, you're waiting for all the data or you're making a mistake at the same time. Is that possible, or is that too much of a reach?
No, I think that's a really good idea, Oliver. And I think we have to do that because so we have a fair bit of traditional sampling data from DFO, but it's specific type. I mean, they're doing a specific type of thing. They're looking for specific species. So we're going to have to restrict our cost estimation of traditional sampling to what they've given us from DFO, and we have to match that against hopefully similar kinds of sampling purposes, or what you're looking for with the eDNA we've got from the teams from you guys. So limiting it to maybe 2 or 3 or 4 objectives, let's call them sampling objectives initially, I think is a great idea to keep the scope manageable, because this could take like the rest of our lives otherwise.
I don't think it's quite that bad, but it's certainly challenging. So Peter and then Margaret, and then we're going to have to shut it down because we've got to move on to the next discussion. Peter.
Just to touch on what Oliver said, I think that's a really, really good point and a really good way to go forwards with the modeling I was talking about yesterday, because those 3 different cases will behave really differently. And I think even if we have maybe a fundamental understanding of a single case study dealing with an invasive species, a single case study dealing with a very rare fish species, and a single case study dealing with maybe a seasonal fish, that gives us a lot of information and a lot of power. And I think there's going to be diminishing returns, at least in terms of the MCDA modeling or that decision toolkit, with trying to have an exhaustive study of every single different fish species, because I think a lot of that behavior is going to depend on more broad characteristics.
Thanks, Peter. Margaret, do you want to round this out?
Yeah, and I like the idea of using some of our case studies to at least get sort of an idea that for a common species, a rare species, just sort of know what the range is. And I think that we probably have a number of case studies either right within GEN-FISH or sort of GEN-FISH adjacent that we can tap into, like a student that I was co-supervising with, Bob Hanner, was using sea lamprey eDNA, and so paired electrofishing eDNA sampling over 100 sites over 3 seasons, 2 years. Like there's a large data set there that can be mined for some of this, and I'm sure like with the goldfish and a few other GEN-FISH projects to at least get sort of a ballpark perhaps.
Yeah, so I think we've got some take-home messages there. I think Tina's point is a good one, that we need to move forward with coming up with solutions. And I sort of charge activity 1 and 2 leaders to remind the SMT that we need to come up with a plan for that. Is there anything pressing for eDNA toolkit before we move on to the fish health toolkit? Okay. And again, if you think of something, you can always pop it in the chat, because I believe that's getting recorded as well. Good. Okay, so now we'll shift gears to the STP chips. Ken couldn't be here. He is teaching. It's his second to last class, and he's got a lot of material to cover, so he couldn't punt on it. I would say that the development of the transcriptional profiling database of genes is doing really well for salmon. It's doing well for some sort of niche fish groups, such as the sturgeons and possibly lampreys. I think, as Nick Bernier brought up this morning, cypriniforms and perciforms are going to be challenging. However, in our proposal, we targeted a group of those species within the list of 45 species. So we don't have to do everything. But if we do that diverse range of species and we have those assays, other researchers who may have their particular favorite minnow would be able to use that as a starting point and modify those primers and probes to, actually work for their particular taxon. But I think one of the real challenges is going from having the data, which activity 3 has made relatively straightforward, to analyze and interpret. The analysis is fairly straightforward, sort of R-based pipeline. The interpret's going to be challenging. And, I'd ask, do we want a tool that's going to look at the data generated by a chip and just pop out saying this fish is stressed by temperature and by pollution? Chris?
I think it's going to depend. I think that the main interest will be aquaculture species and economically important ones. But for interpretation, I think some of these genes will be involved in multiple pathways. So for simplifying, it might actually be good to sort of have a matrix and say, here's the genes we're seeing expressed in concert, and here's the different stressors they're associated with. It may not be possible to give a final answer and saying you've got too much aluminum in the water, or basically narrowing it down to one stressor, but say these genes are all lighting up. Here's the various stressors they're associated with. So be able to say, here's where to focus your efforts and tease out which of these is the causal factor. And John brought it up earlier. It's quite possible it'll be more than one stressor acting. But having basically a scorecard, basically an associative matrix, would help them interpret the results and be more confident if they're actively involved in the interpretation themselves. Just for species, I'd really like to see a plug on the perciforms, because walleye, sauger, zander, I think they're all going to be important.
Walleye's on our list, so. And I
would add, zander's here in North America now. We're stuck with it. Okay. And it'll be showing up in Manitoba with the next good flood. And sauger are likely easier to culture with walleye. But I mean, also several jurisdictions have shifted to intensive culturing methods, which are going to be imposing or are imposing massive selective pressures on what should be a free-ranging top predator. So there's some interesting research questions there, but also some economically important questions to tackle. Wait, sorry, tangent.
Yep. No, it's a good point. And I've had interest in using the chips on wild populations, just to the way it was described to me is to confirm that the populations are stressed. And we're not near that yet, but in theory, the chips should be capable of determining whether it's at a baseline or whether it's off sort of the baseline transcriptional profile. This is something that Nick Bernier brought up yesterday in the workshop. It's going to work really well where you have controls. So you have tanks of fish that are happy, and then you've got the tank of fish, your fish culture system, that aren't doing well. And then you run them both, but you have that control or that baseline. And the question came up, what if you don't have a baseline? You just have a population of fish that are declining and you have no idea why. Can you use this?
Yeah, what's normal? And without a foundation, it's really hard to answer that question.
Yeah, and the idea behind the STP chip was through all of these trials and case studies that we will come up with what is sort of a baseline transcriptional profile for a control fish of species X. And then when you compare your data to the baseline, you can imagine the potential objections to this.
When it's ready for that, I think there's some wonderful opportunities in the Great Lakes and elsewhere. In the Great Lakes, where there's the documented areas of concern, or AOCs. So we know it's a stressful environment. It's usually been due to pollution. And be able to say, how are they doing there versus pristine?
Yeah.
Anyway, sorry. There are some good opportunities waiting in the Great Lakes and some of the larger inland fisheries.
So, general question to people who are interested in the STP chip. Do you think that general adoption for wild populations is possible? Or outside of the realm of GEN-FISH, but maybe possible? Tina.
I think the end users that I work with, they're NGOs, they're government, and they're small communities, and they're interested in how their wild populations are faring. So the only caveat is they really don't want to have anything invasive done. So my question is, I think they would be, again, because you keep saying, look at the profile. What is the profile saying? We know these are usually upregulated, and it's in combination. I think that's where the power lies. And I think, Chris, you're intimating that. It's what is the transcriptional profile that'll tell us something. And if it can be coupled to something that is still informative but non-invasive, I think that's where it might break down a little bit. But Daniel, what are your thoughts? And I was wondering if you could give us an update on how the non-lethal tissue sampling is going for the STP chip. Generally, is blood an option? We had some data yesterday in terms of skin and maybe blood, but how's it going?
Okay, so to give you a couple of quick answers. The original idea of transcriptional profiling is a single biomarker, that is a single gene, is not going to be reliable, but maybe a dozen immune function genes might give you a pattern that you can recognize relative to a baseline. Secondly, the non-invasive, I'm going to let Brian answer that because his student did that. And Brian, how would you characterize the non-lethal?
Again, it's going to be multiple markers, and even then, it'll depend on what's going on. A viral pathogen, you might be able to peg. A parasite, you probably definitely could peg, depending on the way the immune system goes. For bacterial pathogens, they use genes that are upregulated with lots of other things like stress and other things. So that one is going to be, like John said, it could be any one of these several things. But yeah, I think it's. I mean, I've seen papers back in the day using microarrays trying to say this fish was infected with a virus, and they went even and said it was a retrovirus, which was, it was ridiculous back then. I think we know more now, and we will have better targeting on this chip, but I still think the answer will be it depends. It's not going to be cut and dried, and it'll depend on the markers we choose. And as was pointed out, some of those markers are very common to everything, but there might be the odd, like, interleukin 32 that we're not looking to in fish yet that is specifically upregulated in a specific condition. We just don't know enough about immunology to find those things yet. But it may be that there's like a good marker for those things that helps guide us. It'll just take time to figure it. But for right now, it's working okay. It's just, I think the set of genes we have might not be broad enough. If that's an answer.
But do you think it would be broad enough to say it's an immune challenge?
Yeah, I think you definitely say there's immune challenge with some kind of illness going on. And as I said, parasite probably definitely you could peg because it's a completely different set of genes. Virus, there's some specific genes that go up, but bacterial, it's common pathways for almost everything.
Yeah, dog's breakfast.
Yeah.
Yeah. Nick. Oh, you're muted.
Thank you. Tina, I was asking you about the non-lethal samples. So I think gill samples is no problem. I mean, people have been doing that non-lethal samples, for example, of ATPase activity in smolts for a long time, and that's readily acceptable within government practices. You know, when they produce hatcheries and trying to determine quality of smolts and things like that. So, and certainly the work that we've done with repeated gill samples at 3-week intervals shows that the fish are still performing very well in terms of health and growth and all of that. So I think that should be readily acceptable. My own personal belief is that we're still another round of GEN-FISH away from developing chips that would give you the profile of a healthy fish in a wild setting. I was going to give you some insight from some of the stuff that we've been doing, just to kind of put it in the broader context in terms of where we're at in our understanding of where these profiles, what they tell us, and some of the unknowns that we're discovering, in part by using the chips. So we're doing some work looking at the effects of fasting and how fasting. So fish with different levels of energy reserves. So imagine Canadian fishers that go through a winter and that don't feed for 4 or 5 months. And then they get, abundance amount of resources in the spring. So what we're finding out is that, so you fast a fish for a certain amount of time and greatly reduce its energy reserves. Fish are incredibly resilient at just shutting down metabolic rates, shutting down reaction rates. Now you go and you stress these animals. And what you find is that acutely, the fasted fish don't respond. They respond a lot less than the fish that are well fed, right? And so now you have to take energy reserves in this equation just to interpret what the expression profile means. Now, of course, these fish live at various temperatures, and temperature determines metabolic rates in fish. And so the expression profile is going to be temperature dependent. So now in your function, put in temperature, put in energy reserves. That's even before you start to assess the stressors that you're interested in that might be trying to explain what this expression profile might be in a healthy fish and how it might differ. So I'm not trying to be the devil's advocate. I'm just trying to say that we are developing wonderful tools that is giving us some great insight into this. But I think we'll be challenged in the next 12 to 18 months to come up with, saying this is the profile of a healthy fish. That's my 2 cents.
Thanks, Nick. Yeah, that's a good point. Chris, you're up next.
Thanks. Yeah, in terms of your question about would there be appetite for using this in wild populations, I think very much so. I know for working within the M&R, governments, they tend to be reactive more so than proactive. They are very much crisis driven to put out the nearest fire. And a lot of what they're limited to is trying to manage expectations. So changing regulations, that's at least 2 to 3 year process. So even though they're not normally able to factor into basically prognostic sublethal effects, they would very much like to be able to know in advance, how could we adjust harvest pressures or quota allocations to avoid a crash. Walleye in Lake Nipissing have been a nightmare for over 10 years now. The pie shrinks, everybody wants their piece of the pie, and they don't want either the pie or their piece to shrink. The pie itself shrunk. So to be able to have a tool that would let them know in advance, we really need to revisit this, being able to identify what set of stressors are relevant would be ideal. The same will apply for the fish health chip. We are seeing new diseases emerge. You probably all remember there was a massive carp die-off in the Kawartha Lakes. That was 15 years ago now. It's still resonating, and the government did not handle that one well at all. They desperately wanted something to put out that fire. So to have the ability to see in advance or rapid response to be able to say, we have an issue now, what's driving this? To be able to have a quick answer, to be able to get out to the public and say, yes, we are aware, we've identified possible causes. The decision makers would love not to be standing in the middle of a fire before they get this information. So I think there is quite a bit of appetite.
Chris, I agree. And I think part of the reason why GEN-FISH was funded was the recognition that early warning of any kind could be valuable. Nick's point is good, though, that perhaps I may have oversold it, but we got the money.
But he's absolutely right that condition and bioenergetics are going to have a huge influence on what's going on. But that's also something that could be communicated to the end users and say, while you're looking at this, here's some information you want to consider and maybe some extra data. I mean, fisheries biologists talk condition in their sleep. And it's a crude measure, but it's surprisingly informative. So maybe it only gets us 90% of the way to a more nuanced answer. But that takes out 90% of the associated noise. That's pretty good.
Yeah. Thanks, Chris. Brian.
I will add, applying the health chip to wild fish has, I mean, I was still thinking aquaculture. Wild fish is going to be complicated because even more of a problem with wild fish is they're not often infected with one thing, right? They have a parasite and they have a virus. And if you have a bacterial infection, it creates a lesion, then you get a secondary infection. And a lot of the problems, at least what I've done so far in fish, is if they're infected with a bacteria and a parasite, like in mammals, you pick one response or another, right? You worry about the bacteria because it's going to kill you and you put up with the parasite, depending on your life stage. In fish, it tends to just mix up and they have a chaotic half response to both, and it doesn't work either way. But in terms of a chip signal, that will just look like a big chaotic mess. So in wild fish, you might just say, look, there's a fish health problem here. You're going to have to dig into it further, right? As I said, unless we can find these odd, weird genes that are expressed in weird ways. But it's, I think, like Nick said, we're a good generation of GEN-FISH away from having anything definitive for wild fish. It might work for aquaculture fish because at least we have a handle on the pathogens there, but for wild fish, it's going to be tough.
Yeah, I agree. I'm already thinking about regen fish. Oliver.
Yeah, I mean, I'm going to just suggest something exactly what I suggested for eDNA. I mean, like, I don't think that the promise of this was overreaching, but, you know, I mean, these things are complex and organisms have enormous amounts of capacity, you know, to sort of deal with acute and chronic stressors and still do quite well. And the ultimate goal is to measure performance and fitness. That's the only real way for us to be able to tell whether these organisms are failing or doing well. And that takes a lot of work and a lot of context to figure out. But I mean, if we chose a couple of systems that were in extremes, so a fish that people care about, that's in a declining population, same species in a population that's neutral or doing quite well, and we can show so much energy is being put into this and the animal's failing, I mean, that to me is a win, you know, and for a 4 to 5 year grant, that is a major win, and especially if it's in a wild system. And then we can build off of that and build in all the nuances and the complexities for all those other species that we care about. If we can do that, I think that's phenomenal in 4 to 5 years, given what the goals are and what all the complexities and hardships are doing this kind of stuff.
Yeah, I agree. And I think we may be picking at straws. I don't know, is that the right term? In that I think we all agree that a 56 gene chip with the proper selected genes that we're getting data for and refining the list will let us know if the fish is in trouble. Whether or not it's going to be able to tell us what the stressor is, that might be Gen X fish or next gen fish. But I think it will allow us to identify whether a fish is showing a coordinated gene expression response or a complete breakdown, like just totally uncoordinated up and down regulation, or something that's sort of within quote unquote normal. And to Oliver, your point, I think the data we're going to have most for will be the salmonids. And it's no coincidence that the salmonids is what most people really care mostly about, with the exception of walleye and yellow perch and sauger. But so we're running out of time for this. I have to admit that coming into this AGM, it had never occurred to me of the logistic and technical challenges of applying the STP chips to wild fish. I kept sort of thinking about captive fish and then just sort of extrapolating without sort of, wait a minute, you're not going to have a baseline, you're going to have multiple stressors, you're going to have environmental differences that aren't even perhaps measured. So I think that's something that we can take up in our discussions when we go to regular sort of lunchtime meetings about activities 3 and 4. So before we move to activity 5, are there any other pressing questions or ideas that we could put on the list?
I think it's a worthwhile, Daniel, to say, okay, when we're collecting wild fish, we have them in our hand anyways. Get a length measurement, take a photo, take a, you know where you're sampling from. You've got historical records on temperature. You've got historical records potentially on the community diversity. So when we're analyzing it, you're going to look at chip results, but you have to put it in a model with all these covariates. And I think that might be something that we would have to have as a caveat before next gen fish comes into play. So it's interesting consideration. And Chris is already saying you can get condition off of length and weight, you know? And they're like, so you don't need the STP, but you might want to see if you could like help in some of the stressors, like the energetics that Nick was talking about.
Yeah. And it's interesting how the two toolkits are sort of merging into this. We need a functional response model that includes more variables than perhaps we originally thought of for both eDNA and STP-chip interpretation. And the last thing before we switch that I wanted to answer, Tina, you brought up the non-lethal sampling, and Nick mentioned like a gill biopsy. There's also been some very promising work on a blood sample, and my personal favorite was skin mucus. And Brian Blandine's data on the mucus looked very promising.
I was actually, Nick, just talking this morning to Cat C, the company I work with in PI, and they've been working a lot on using mucus for markers and how to preserve it and how to collect it and stuff. So I asked them actually if they wanted to use some of these chips. So I think that has some promise, I think. So
yeah. Now the eRNA, I haven't seen Shahin's data, but my fingers are crossed that again, this would not be, I don't think it'd be valuable for wild populations. They're just not dense enough, but certainly for culture, they might be dense enough that you could sample the water and determine whether or not the group of fish in that tank are expressing stress genes high enough to be exuded.
Oh, look, all that COVID in the wastewater thing was all based on eRNA.
Yeah.
I have my questions. They even ditched their reference gene. They just gave you the raw reads and
yeah.
But hey.
yeah.
Theoretically, it works.
Yep. Theoretically, yeah. Good. Okay, so what I'd like to do is hand this over to Tina and John and any other Activity 5 folks that are still there to lead a discussion on Activity 5.
Yes, and Amy's still with us too, and Peter. And so my biggest push always is to have our activity members reach out to everyone else on the team to get connections to those they can interview, they can sit down with, they can pick their brain. Because remember, the wheelhouse is in resource economics, it's not in transcriptomics. The wheelhouse is in animal ethics or ethics. It's not necessarily in eDNA. So the types of questions that we can pose to activities 1 through 4, we might not be able to. And so we need to have activity members 1 through 4 tell us what are some of the considerations, what are some of the questions. And then we also need access to our end users. So we need both. I don't want to speak on behalf of Activity 5, and that's my interpretation, but we really do need access not just to those of us here at the AGM, but to our wider population at large, or end users. So I'm going to leave it over, though, to Amy and to John and Peter, see if there's anything specific. But just to let you know, that's why I'm constantly making calls. Okay, let's get together. Let's have a workshop. Let's have someone sit in on a workshop and have the communications going back and forth. Look, these are some of the considerations that a fisheries manager might be interested in, and an ENGO might be interested in. Then it's like, okay, these are the types of questions. Can we get access to an ENGO? Can we validate this with someone who works in an NGO? So we need to help facilitate the connections and the networking so that the questions are being properly asked by multiple parties. So this is what I want to be able to go forward with our last year and a half of our funding, just to make sure it's done. But that's my broad generalizations. But Amy and John and Peter might have much more specific requests. Yeah, go ahead, Daniel.
We have like 21 end user initiated applications of the STP-chip. I don't remember them. Some of them are wild populations. Most of them are captive. But I wonder whether a connection between those end users, those specific individuals who are working on that, might be valuable.
Do we liaise with Paige to figure out who these are? Like, who would be the best way to connect us?
My guess would be Paige and possibly me, leading to the PI, and then through the PI, because they typically have the sort of the personal connection to the non-academic partner.
Gotcha. Okay, thanks, Daniel. So you're saying these are our end users that have requested specific uses of both eDNA and our STP chip?
The 21 that I list were all STP.
Okay, so we would probably need the same for activity 2.
Yeah, those are the case studies.
Sorry. Thanks. Amy, go for it.
I just wanted to say, like, thinking ahead, I appreciate the conversation that we've been having about looking forward. And it's occurred to me as I'm analyzing the focus group data that it was necessary to set them up the way that I did with various stakeholder groups, but it really is just kind of, scraping the surface. And so if there is a subsequent GEN-FISH project, whatever it's called, I think it would be valuable to take those specific stakeholder groups that I've done the focus groups with, and then do more intensive work with them. Because at the end of the day, I'll be able to say, okay, here's what came out in the focus groups, but there might be, I think there's richer data to be had there. So just something to consider for the future.
Thanks, Amy. Can you give us an idea of the stakeholder groups that you've already connected with?
Sure. So
who's missing? Maybe who's missing too?
We had good representation from recreational fishers, the public, environmental and animal focused organizations, researchers, some government agency staff, but I think it would be valuable to have more if possible, and as well more on the commercial side of things, aquaculture. I think that would be helpful, you know, because the representation that we have right now isn't optimal. And then we're currently engaged in relationship building, and we've drafted a research contract with an Indigenous community here in Ontario. So we'll have, I expect, really valuable information from that engagement as well.
Amazing. So I'm just writing notes here. So again, it might be worthwhile for us to connect to Paige, then to connect to our co-PIs who are working directly with industry to see whether or not it'd be possible. So any kind of user groups that you feel that you need to target more and more, Amy, let's have a discussion then with Paige, and then maybe we can facilitate some of the networking happening there too. So you can fill it in, and like you also said, have a more in-depth conversation. So this is good. Stay tuned. John, do you have any, sorry, Amy, was that good? Does that capture what we can do to help you?
Yeah, I was just going to say that would be helpful because, you know, I've reached out to these groups, but just haven't received response, right? So I understand people, you know, are busy, limited time. So maybe if people, you know, members of the team have relationships already built with some of these constituencies, I think that would be helpful if we could leverage that instead of me, you know, cold emailing people, because for some of these groups that just hasn't really panned out.
Yeah. Thanks, Amy. We're going to help you out. Don't worry. How about you, John? What do you need help with?
Yeah, we need to, so really the main thing left to flesh out in our cost model analysis is we need to talk to somebody who can give us a broad overview, walk us through what happens after the sample is taken and stored in the field. You know, it goes back to a lab, presumably, but we're going to need like a fair bit of information on the possibilities for processing the sample. For example, the end user who's on the end user committee, and Peter, you'll have to remind me of his name because I can't remember, who we were talking to, told us he's a consultant, environmental consultant. When they do eDNA, they purchase a kit from a company that supplies them with everything they need to take the sample, filter the sample, and then it gets sent back to the lab. The lab processes, gives them the results. I can't remember how much he said it costs, but that's obviously one option for, you know, figuring out the cost of processing the, you know, the post-field work processing cost. But I don't know. The question is that's presumably very specific to what they were looking for. So is who in our team should we talk to, Peter and I, that can give us sort of the broad overview of what happens after the field sample is taken, and then we can maybe go and talk to some specific end users to get, you know, details about market rates for processing the samples and so on. Yeah, so question one is, who on the team should we be talking to about what happens after the field sample is taken? In practice, I don't mean in our research.
Don, I would say there are
Margaret,
a bunch
Daniel.
of us. Pardon? I think a bunch of us could
Margaret
do that.
or Daniel, would you be able to?
Yeah. The issue you're going to run into with somebody like Margaret or I, or Mark or whoever, is we know how much it costs to do it for research, where it's basically, this is the actual cost. And I know that environmental consulting can have markups of multiple times, like not just double, but triple or even 10 times. I have no idea what the markup would be for contract eDNA. I've never looked into it. I wonder if you could just find it on the internet.
Probably could, but it might be very specific to a very specific type of objective. And we will probably need to know a little bit more about that. We can certainly go to the internet and search on the market, but probably talking to you and Margaret will get us started anyway.
Or Matthew, yeah.
Yeah, because I suspect that sort of thing would be maybe the upper limit, that if you were doing it completely hands off with no collaborators, that would be maybe what the upper cost would be. Presumably only really feasible for somebody who has fairly deep pockets, or if they have relatively few samples. And then, talking with Daniel or my group, that might give us an idea of how much it would cost with a collaborator. Because so we've worked out costs. I won't put Jesse on the spot right now, but Jesse in my lab has been really good with working out costs with, or maybe they're willing to be put on the spot, like in terms of, if we have 100 samples, this is roughly what it is per sample. If there's 1,000 samples, this is roughly what it is, sort of thing, whether we're factoring in salary or whether it's a student project and we're not factoring in salary. So that I think, the first few times I asked you to estimate costs, Jesse, it probably took a fair amount of time. You've got your spreadsheets now, and I think you're able to do that much more efficiently. There's not one single answer. It's not like running one sample is going to be the same cost per sample as 1,000, but we've got some of those things.
Yeah, usually updated about every 6 months, just because the cost of all of the reagents fluctuates pretty dramatically depending on what is sold out and what is high in demand. Particularly with COVID, some of the products were high in demand for testing, so things were, very variably priced. And it also, as we talk about all the time, it's going to be also dependent on how many species you're testing, if you're doing all of those species for every site you take, for at least for the single species assays that we do.
Yeah. So again, not to volunteer you or anything like that, but I was thinking yesterday during Peter's presentation that for refining some of the costs, you might be able to give some insights.
Yeah, I definitely can do that.
That'd be helpful. Thanks, Jesse.
Thank you both.
Definitely be contacting you. I know that I don't know, you've probably heard of Paul Hébert, one of our University of Guelph superstars. He's talked to me about the cost of well, okay, let's just leave it at that. The cost of processing for what he does, which is identifying a species with the barcode in China, is like he just can't even touch it, and he's able to do it for about $5 a sample or something ridiculously low. And I know from talking to my daughter, who's a molecular biologist, that sometimes the lab she works in sends their stuff to China to get the whatever it is getting processed. I'm an economist. She's a molecular biologist. I try to understand what she's saying.
Yeah, I certainly can't compete with $5 a sample.
Well, it's very specific, I guess, what Paul Hébert's group is doing. It's very specific, so they get very high throughput, and so it's all very automated now. So, but the Chinese can do it for even less than that, apparently. So anyway, it just seems to me like it's all over the map. So it's going to be very dependent on, as Jesse was saying, what it is you want to do. And I assume not much of it will be really standardized for this kind of testing.
Yeah.
And John, I know that, oh, sorry, Jesse, I didn't mean to interrupt.
No worries, go
ahead.
for it.
I was just going to make reference to, there are a couple of kits that Chris put in our chat, and there's again a two-tiered cost structure as well. So like you said, high throughput, number of species. Sorry, Jesse, please continue. I didn't mean to cut you off.
No, I was just going to agree with John that as we've talked about, like depending on your goals and the scope of the project and how much like in-kind, like support your collaborators giving will vastly fluctuate the cost it will take.
And Daniel, before you ask or answer a question, could I just pose one more? Just curiosity, when you get these samples, well, you're testing them, you're running the samples yourself in your own labs, but if a consultant were to do this, are you asking for like bioinformatic interpretation of the results in addition to just like getting the raw data from? Like, are you paying for somebody to actually give you an interpretation of the results as well as just sending you back raw data? Because I assume that's going to make a huge difference in cost.
Good question.
It just struck me that University of Windsor's environmental genomic facility provides eDNA analysis on a fee for service. And so Shelby actually has a price list. And if you want it interpreted, she has a cost for that. And there's sort of a minimum number that she'll do, and then it's by plate, so by hundreds of samples.
That's great.
Daniel, did you want to add anything or have your own question? Or John, was that good? Was that a good
Yes.
pathway to follow?
That's good, thanks.
Right. Go ahead, Daniel. You're muted. Was that it, Daniel? Was that it for you? Because I'll go to Matthew. Matthew did have his hand up.
I was
My
just going to
point was
say
the
that
one
I was just on the side looking at costs for eDNA, and most of the websites are very coy about giving costs. They don't have it sort of like Canadian Tire. So it's not as easy as I thought it might be by going online. That was my point.
Gotcha. Okay.
The only thing I was going to say was just that Shelby at the EGF was a great resource for that, or a potential resource for that. So Daniel said the exact point that I was bring up.
Excellent. Awesome.
Yeah, because the more I think about it, I mean, it's not even just a matter of scale, whether you're running 100 or 1,000. Do we have the primers and probe already in stock for that species, or do I have to order, primers and probes for a particular species to get started? So yeah, that there are things that need to be taken into account.
Yeah. And John, Jesse's given you their email address. That's so kind of you, Jesse. Thank you so much. And Brooklyn has said whether the assay exists and is in stock needs to also be designed or ordered is a factor. So just confirming what Margaret said. So a lot of things to take into account, but hopefully we could end up on a ballpark figure anywhere between $15 a sample to $30, contingent on the following, rather than getting these exact metrics. Excellent. Is there anything else that our Activity 5 group needs? Any questions from our non-Activity 5 members? I'm supposed to say the alphabet in my head. That's how long you're supposed to wait for questions. But I could race through the alphabet really quickly, so I have to do it backwards.
Yeah.
Brooklyn,
I have a question,
yes,
Tina. It's Brooklyn here. So I've had some I guess companies reach out to me at conferences and stuff. One that comes to mind is, and I'm blanking on their name now. I have it somewhere. Oh yeah, I'll bring it up anyway. But I have his card somewhere. But they, there are a lot of companies that don't necessarily, they just supply products, especially eDNA products, and they have they'll sell the Smith and Root backpack eDNA sampler, and then their clients will ask, well, where do we get the eDNA samples processed? And they don't have anywhere to send them for processing. So is that something in this conversation that we're having? So I have my niece here. She's a toddler, so she's distracting me a little bit. So maybe it was mentioned already, but is that something that we're planning on doing? Because I do have a lot of contacts or people who have asked after seeing my posters whether we are looking to contract our services out.
Daniel, what is the stance on that? Good question, Brooklyn.
GEN-FISH isn't. I think Genome Canada would be thrilled if we had a spin-off company, but the EGF is doing it. And I think that if it became more and more subscribed to, that there would be money to hire technicians and deal with it. So so far, the eDNA contract work through the EGF has been relatively minor, associated with a couple of consulting firms.
Yeah,
Yeah,
I
that's
would
good
say
to know.
yeah.
Yeah, just
So I would
'cause,
say
like,
at University of oh, sorry, go ahead.
No, go ahead, Margaret.
Sorry. Yeah, I would say at the University of Manitoba, if we were wanting to do that, the University of Manitoba would be wanting a much higher overhead than they take on research funds. That anything that we do now is collaboration, but yeah, no, they'd be wanting a much higher overhead if it was even possible.
Yeah, just knowing that the EGF does it is really good, and being able to redirect to them would be great. Because yeah, I didn't know of anyone other than consulting companies, and from what I've seen with them, what we talked about yesterday about the proprietary assays and stuff like that, I didn't feel comfortable sending them to any of the consulting companies that I have seen advertising eDNA. So I've worked, with Shelby a little bit, and I definitely trust her work. So sending to the EGF would be good. 40% overhead. Oh, okay, well.
I can add to that from my days in the research office at Guelph. Our Advanced Analysis Center also does external work, and they have a differential fee structure for external versus internal. So the overhead is kind of built into the fee structure, and they do kind of probably like most university DNA labs, they do kind of boutique work, not sort of high throughput stuff. But they're very capable of doing just about anything, I think.
Yeah. I just want to make sure before I get to you, Matthew, that Paige has said previously that she's been told by American colleagues that there are fewer labs in Canada versus the US that aren't associated already with a government agency or university. So the fact we do have environmental consultancy that can do that, but we don't really know too much about that. But do you, Matthew? What were you going to say?
All I was going to say is that, just to echo the point about the EGF, I mean, recently, I think the EGF is actually probably pretty reasonably competitive relative to a lot of other environmental consulting firms for doing this type of work, simply because they have the infrastructure and techs in place, and the eDNA samples are kind of an add-on of other work that's built into the EGF, because it's kind of like a general purpose sequencing facility. They can do a lot of different things. I know that Banff, I have some colleagues at Banff who are doing a lot of salmonid eDNA stuff because they're rote-owning lakes, and I pointed them in the EGF direction. And my understanding of this situation anyways is that they're mandated to go out and solicit quotes from like 3 or 4 companies as part of a bidding process. Like that's the rules in Parks Canada, that you have to do that. You can't just give a company a contract, and EGF was the one that they ended up selecting to do the work. So Shelby's processing their samples right now for their latest rote-known efforts, because they're using eDNA to detect, see if they eliminated the fish in those systems. So I think the EGF is probably pretty reasonably competitive. But
that's really good to know.
Sounds like it's time to raise the rates then.
Margaret, I was thinking exactly that.
Banff is going to be
I know,
pissed
we
at me
get
now.
chosen. Yeah. I know. Thanks, Matthew. I don't think there's any more questions then for Activity 5 or from Activity 5 members. So Daniel, Margaret, I'll just pass it back to you as our stalwart leads of GEN-FISH version 1.
I like the version 1. Well, I don't have a lot to say in closing. I appreciate the effort everybody made to be sitting in front of their computer for so long, over 2 days. I think some really important points were made. I think that all of the activities got some direction that will be valuable. And I think we also managed to sort of highlight the fact that we need to get what we can done in the next 18 months. And I think we're across all of the activities, I think we're in a good position, given the fact that we started out with such a strike against us. It is remarkable what I saw, yesterday and today in terms of the progress we've made. And even if GEN-FISH did get closed down January 1st, if Joe hadn't managed to get us a 18-month grace period, I think we would have real deliverables to show for the 4 years we've done. Margaret, would you like to add to that?
Yeah, I would just reiterate that I have to say, even though it's not as enjoyable doing it virtually, I think we covered a lot of territory over the past 2 days, and it was really nice to be able to have so many people participating. So it definitely was very worthwhile. And thank you, Paige and Joe and everybody, for all of your efforts to pull this together. It's been great. And yeah, that the proposal was overly ambitious. This is the first time that I'm starting to see that we might actually be able to deliver most, if not everything. So yeah, no, it's really really gratifying. Sorry, I just have a tickle in my throat. I'm not getting all teary about it or anything.
Emotional, but yes, actually, very good point. I'd like to really acknowledge the contribution of Joe, Paige, John, Keta, a host of undergraduates who've been working behind the scenes supporting GEN-FISH, and of course the HQP, where we would be nowhere without our students and postdocs. So, and I just want to, remind us all that next year's AGM might be more pleasant. Matthew.
Daniel, sorry, go ahead.
Incorrigible?
I just think you're going to get robbed. I think you're going to have such a horrible experience that you basically set yourself up for a horrible vacation. Yeah, anyway, okay. Matthew, what were you going to say?
I wasn't going to say anything. I must have misclicked or something like that.
Dan, are you telling us the book now? Is that what you're saying? And I just coded to GEN-FISH?
I'm sure that Joe will take care of that for you, Nick. Anyways, thanks so much for everybody and for joining us. And we will probably come up with a document to summarize what we came up with this. I think it'd be valuable for our upcoming ROC report and also valuable for all of us moving forward. Thanks so much.
Thanks for reminding us of the ROC report.
Turks and Caicos, ROC.
Have a good winter break, everybody, and happy holidays, happy restful times.
Yes. Yeah. Great seeing everybody. Yeah.
Yeah, it was great.
Bye, everyone.
Bye.